ये एवरी लॉगिन को क्लिक करने का नहीं ना स्पॉन्टेनियसली।
Practical var dedi. Yüzü tiye likhit है ना ये थे लिखे ये लेकर से तक ठीक है तो करता हूँ याद हूँ यूज चीज ना याद है ऐसे नहीं रिक्वायरमेंट याद लेकर वही ऐसा कि ना याद लेकर ना name of the employees याद लेकर number number को close number आपका तक रिक्वायरमेंट से लेकर
ये बोल रहा करने के लिए क्या है एक पेंसिल बनाने के लिए
अलग आ सकता है
मीटिंग वाला नहीं से क्लिक करो
तो साइड के लोग बहुत थे कश्मीर में
ये अटेंडेंस है इसमें
और समोसा का काम जो है ना समोसा हर 1 किलोमीटर में समोसा है ठेला भी और नाई जितनी सब बेचती
नाई है या फिर ब्यूटी वगैरह करना है
वो लोगों ने वो वहां पर गोली वाला दौर था तब भी वहां से हटे नहीं जब मॉल खराब था और उनके ताल्लुकात बहुत अच्छे हो गए पुराने हो गए ना बोल कर दूँ मैं
कश्मीर
अब कर दे
अब करने में क्या
वो उसने जो दिया है ना भेजा है ना जो ब्रोशर भेजा है नहीं उससे पहले जो भेजे हैं लिंक भेजा है वैसे नहीं उसे भी क्लिक करी तो क्लिक करके रखी थी मैंने लाइक स्टार्ट हो गई नहीं मैंने रजिस्टर नहीं की
Assalamu alaikum and a very good afternoon to everyone. Myself, Dr. Kausar Fatima Firdaus, Assistant Professor, Department of Ilmul Khabalat, Wamraz and Iswan, National Institute of Unani Medicine, Bangalore, your host for today's webinar on emerging technologies in healthcare. We are elated to have you join for this exciting session where we will explore some of the groundbreaking innovations shaping the future of healthcare. As all of us know, the healthcare industry is undergoing a significant transformation driven by advancements in technology that promise to enhance patient care, improve efficacy, and reduce costs. From artificial intelligence and machine learning to telemedicine, blockchain, and wearable healthcare devices, new technologies are helping healthcare professionals provide more personalized, accessible, and effective treatments. In this webinar, we will delve into how these innovations are already making an impact and what the future holds in terms of possibilities. For the formal beginning of today's webinar on emerging technologies in healthcare, may I now request Professor Sayyad Shah sir, Director, National Institute of Unani Medicine, for welcome address. Over to you, sir.
Thank you, Dr. Kausar Firdaus. Assalamu alaikum. Good afternoon, everyone, and welcome to today's webinar on emerging technology in healthcare. The collaboration of emerging technology with the Unani system of medicine offers significant potential for advancing and integrating traditional and modern healthcare system. Unani system of medicine is an ancient indigenous system of medicine based on the healing principle of ancient Greek medicine and further developed in the Islamic world. Focuses on the holistic approaches to health using natural herbs, dietary particles, and lifestyle modifications. Emerging technologies such as artificial intelligence, biotechnology, data analytics, and digital platform could help strengthen and expand the reach of Unani medicine in several ways, including standardization and quality control, research and development, clinical trial and data analytics, personalized medicine, education and awareness, global integration and collaboration, sustainability and environmental consideration and integration with modern healthcare system. The collaboration between Unani system of medicine and emerging technologies can unlock the potential to create a most robust, scientifically validated and globally accessible healthcare system. Integrating Unani medicine and technologies could lead to new breakthrough in holistic health practices, personalized care, and evidence-based treatment, improving patient outcome and contributing to a more sustainable, efficient, and globally inclusive healthcare environment. Thank you once again for joining us. We hope you find today's session insightful and engaging. Thanks one and all. Thank you.
Thank you, sir, for joining us today for in spite of your busy schedule and making this beginning of the webinar very, very awful. Thank you once again, sir. We have an incredible lineup of expert speakers who will share their insights on the latest trends, discuss the potential challenges and opportunities, and provide real-world examples of how these technologies are revolutionizing the industry. So without further ado, let's dive into the fascinating topic and explore how emerging technologies are transforming the healthcare landscape. We have with us Dr. Amy Steele, Associate Professor, School of Public Health, Australian Research Consortium in Complementary and Integrative Medicine, Sydney, Australia. Hi. Is the screen visible?
Hello. Thank you very much.
Madam is Associate Professor in School of Public Health, Faculty of Health, University of Technology, Sydney. She is postgraduate certificate in Health Economics, University of Aberdeen, Doctor Philosophy in Technology, Sydney, Master of Public Health, University of Queensland, and graduate certificate from University of Queensland. Madam has professional representation in WHO, World Naturopathy Federation, Osteopathy Research Alliance, among others. She has conference coordination in over 10 conferences and editorial responsibilities. She is a senior editor in BMC Complementary Medicine and Therapies. She is editorial board member, Integrative Medicine. She has Guest editorial board member in Advances in Integrative Medicine and the Journal of Alternative and Complementary Medicine. Madam has funded research projects over 8 active, and she has completed more than 15 funded projects. And her publication goes on with 246 journal articles, 3 edited books, 5 journal special issues, 44 book chapters, and 6 reports. And the list is to go on. I welcome you, ma'am, once again on behalf of National Institute of Unani Medicine, Bangalore. Over to you, ma'am.
Thank you. Thank you very much for inviting me along and allowing me to be part of this auspicious event for the National Day of Unani Medicine event, workshop and webinar. Please let me know if you have any issues hearing me. I know I'm coming from Australia, so we may have internet issues, as happens sometimes with webinars. But I will just share my screen and my presentation. Is that working okay? Can you hear me okay?
Voice is a bit not clear, ma'am. Your screen is visible.
Okay. Is it, yeah, my screen is visible. Can you hear me now?
Yes, ma'am.
I will try not using my headset and see if that's a little bit better. I'm not sure if that will help. Okay. Okay, does that make any difference? Can you hear me a bit better
Yes,
now,
ma'am.
or is that not better?
You're audible, madam. You can continue.
Hello.
Apologies, I'm not sure what happened there. Can you hear me okay?
Yes, ma'am. Audible.
Hello?
Audible, ma'am. You're audible. You can proceed.
Excellent. Oh, wonderful. Glad to hear it. Okay. I will share my screen and we will continue. And you can let me know if there are any issues with that. Excellent. Okay. Share. Okay. All right. So thank you very much for having me. I really appreciate the opportunity to be a part of this conversation, particularly around emerging technologies, which I believe is an important topic for traditional medicine. And so I'm going to start the conversation by talking about maybe some issues that we need to think about in the context of traditional medicine for any emerging technologies as they develop. Okay, so I'm not audible. So I'm going to talk about the some of these issues that we need to think about when we're transitioning traditional medicine into emerging technologies and how that works. Am I audible now?
Yes, ma'am. You're
Better?
audible.
Excellent. Okay. Excellent. Thank you. All right. That's good to hear. I think we've come up with a solution. So just to orient those of you, this is the research group that I'm from, the Australian Research Consortium in Complementary and Integrative Medicine, is a research group that focuses specifically on critical of traditional complementary and integrative medicine. There's a whole wide range of topics that we cover in our group, and the work that this particular presentation is based on one of those streams of research that we've been working on. So the research group itself is very widely published. We've attracted a lot of competitive research funding. We're actually rated the #1 TCIM research group in the world. And we've been very successful at attracting large government fellowship grants, and we lead a lot of practice-based research networks, and we've got a lot of international collaborations. So I'm really excited to have an opportunity to share this research with the Indian community, particularly within the Unani community within India. So what I'd like to start with talking about is this concept of emerging technologies in healthcare. And the reason I want to talk about that is that whilst we obviously have new technologies emerging, the concept of new technologies is actually not only for modern medicine. It's not only new, and in fact, the traditional medicines that we're all a part of, including Unani medicine, has evolved emerging technologies over time. New uses of simple tools crafted from natural materials, moving into the Industrial Revolution when technologies using steel and glass started to be used. And of course, there's a technology boom that happened, the use of diagnostic radiology, and then, of course, more recently, the digital age, in which we've had much more sophisticated, compact, and interconnected technologies. The thing is, these things haven't happened. Traditional medicine has had contact with each step in this evolution of technology for medicine and healthcare. It's just that they've been used in a particular way and in the particular context of traditional medicines along the way, with us identifying how they're appropriate and in what ways they need to be adapted, if at all, to suit the context of traditional medicine. And the reason for that is that all healthcare, all medicine, including traditional medicine such as Unani, they evolve over time. The difference between biomedicine, which really is not at all attached to its historical roots, it's always seeking new methods for diagnosis and treatment, and disposing with past methods. Traditional medicine systems retain a strong connection to their roots, philosophies, principles, and practices. And so apologies again. Obviously, the Australian internet is causing difficulties today. I will continue on if you can hear me. And so what I'd like to talk to you, sorry?
Screen share.
Is it okay? Can I continue?
Screen share.
Yeah, screen share. Oh, my screen share. Thank you for the reminder. Sorry, I lost my screen share when it kicked me off. Okay. All right. So what I'm going to talk about today is a framework that we've developed that helps to bridge traditional knowledge with contemporary needs, and how that framework might be able to be applied to traditional medicine, to the use and integration of emerging technologies for traditional medicine. So this framework was developed based on a systematic review, and the systematic review looked at how traditional knowledge was being used in contemporary contexts, how it was being used in research, education, policy, and practice. and the way in which it was being adapted and applied for modern day. Because what's interesting, what's different for traditional medicine practitioners is that these historical practices are not historical artifacts. They're not moments in time from hundreds or thousands of years ago that historians might research. They're actually modern practices that are
Internet is very poor, I think.
okay. So so we undertook a systematic review, and the systematic review was designed to identify how traditional knowledge was already being used and how this use might be affecting I'm going to turn off my video just to see if that helps a little bit with the internet. How this traditional knowledge was already being used in these 4 different contexts in research, policy making, education, and practice. And we really wanted to know in what way it was being adapted, this really great framework from implementation science, which is all about adapting evidence into new settings and new contexts. And it has 4 steps. It's called the EPIS framework, EPIS, exploration, preparation, implementation, and sustainment. And it's the implementation and sustainment part that we're most interested in for emerging technologies. But what we looked at was the whole range. How do you identify a traditional knowledge that's going to be used in modern day? How do you prepare it in the modern context compared to how it was prepared in the traditional contexts? How you're actually going to implement it and use it, and how are you going to ensure its ongoing effect? So we looked at it from that particular model because this particular model was developed for evidence-based practices that have been found to work in one setting, one country. I'm so sorry. I'm so sorry I keep being removed. I'm not sure if you would like me to still continue or if it's too difficult to follow the presentation. What would you like to do?
And we would like to continue.
Okay, no problem. Thank you for your patience. Okay, so we use this EPIS framework to evaluate how information was being used based on the current evidence. And based on that, we evaluated the quality of that process of its use. We looked at the characteristics of the articles, the context that traditional knowledge was being used, how people were evaluating if it was good traditional knowledge before they used it, and different criteria they used to make the decision about using it. And if you think about this in the context of emerging technologies, this is really asking the question of how did they decide which traditional knowledge or traditional medicine practices to combine with an emerging technology, and how did they determine that it was an appropriate traditional medicine or practice to combine with the emerging technology. So we appraised the manuscripts based on how well they described their choice of traditional knowledge and their method of selecting the traditional knowledge. And was it actually described, or did they just make a passing comment about it being a traditional practice from a particular traditional medicine system, but also the authority of the individuals who are analysing the text. So did they have training in the traditional medicine system from which the knowledge arose? And did they have experience in the context from which they were going to then use that traditional knowledge, such as were they researchers using it in research or policymakers using it in policy? What we found from that is we found 54 articles, and probably the most important points I want to make the point of here is that whilst a number of them appear to have authority on the condition, on the tradition and on the context, very poor reporting of how they went about selecting the traditional knowledge to implement was noted, and that was not clear enough. It wasn't reproducible, and so they weren't treating their selection of traditional knowledge like they were treating the systematic review, where they would go and select data from a systematic review. It wasn't clearly laid out with steps and methods and justification and inclusion and exclusion criteria.
Voice is not clear.
Can someone tell what is happening?
All right, I'm back again. So the what we found was most of the papers that reported the use of traditional knowledge talked about how they found the knowledge, the exploration part. They talked about how they prepared it or made the decision about which one to use, but they provided very little information about how they actually adapted it to implement it and tested its effectiveness, and also how they evaluated that it was working in the end. So this implementation and sustainment was very underrepresented in the data that we found. And so we then went to speak to some representatives from a whole range of traditional medicine systems within Australia, and asked them a little bit more about how Okay, I'm back again. Yes, I apologize for the technical glitch with my speaker. It's obviously an issue with the internet in Australia. So through taking the stakeholder World Cafe Forum process, which is a consultation process that we went through, we identified a whole range of potential topics. And from those topics, we then went on to develop a Delphi panel. And we went to an international group, went through 2 rounds, reached out to a range of experts. And from that, we developed a criteria. And this became the SITE framework, the Contemporary Implementation of Traditional Knowledge and Evidence Framework. The framework has 3 different categories, or 3 sections. One is guiding principles, 2 is the critical appraisal criteria, and 3 is application criteria. And the purpose of the framework is to direct how people can look at implementing it in contemporary modern uses. So this is perfect framework for people developing emerging technologies for and with traditional medicine and traditional knowledge to make sure it comes together in a way that still reflects the values of the traditional medicine system. So the SITE framework has, for each criteria or guideline, it has a header title, it has a set of statements or questions, and it has an explainer for each one. Now, I'm not going to go through the full document, but you can access it via this QR code here. But I will say, but I will just provide an overview of the different sections and then talk about some examples that are relevant to emerging technologies specifically. So the first section is the guiding principles. The guiding principles talk about the importance of accountability in the translation of knowledge, which means that when translating or implementing traditional knowledge into contemporary use, we have to be accountable for the processes we use, the outcomes we achieved, and the potential impacts of implementation. In terms of important foundational assessment of traditional knowledge, is a second guiding principle. And that means that we have to undertake the foundational work of exploring
There are some technical issues. We'll shortly rejoin. Apologies for the interruption. Madam is unable to connect from Australia. So we'll move on to our next lecture on onco nanomedicine, epitative tool for modern therapeutics. Nanotechnology in medicine has opened new frontiers by enabling more precise targeting of the cancer cells while minimizing damage to the surrounding healthy tissue. This approach can improve the efficacy of treatments such as chemotherapy and radiation therapy, and also facilitate the delivery of drugs directly to the tumor site, reducing side effects and enhancing therapeutic outcomes. Our next lecture on onco-nanomedicine, innovative tool for modern therapeutics, refers to the emerging field of cancer treatment that combines nanotechnology with oncology. It explores how nanomedicine, which involves the use of nanoparticles and nanomaterials, can be harnessed to improve the diagnosis and treatment and monitoring of cancer. For this, I invite Dr. Professor Zeenat Iqbal ma'am, B Pharma, M Pharma and PhD, Professor, Department of Pharmaceutics, School of Pharmaceutical Education, Spur, Jamia, Hamdard. Madam is Professor, Department of Pharmaceutics, School of Pharmaceutical Education and Research, Spur, Jamia, Hamdard, New Delhi. She's a postgraduate and doctorate in pharmaceutics from Jamia Hamdard, involved in academics and research activities at the Department of Pharmaceutics per Jamia Hamdard, a proactive member for the teaching fraternity with a penchant for quality education and research, an active research supervisor with H index of 4, special interest in student-centric co-curricular activities, and able organizer of both scientific and non-scientific programs, and major contributor to the university corporate life. Madam has to her credit over 42 awards. She has been a resource person in 56 lectures. She has presented papers in 41 conferences and attended 33 invited lectures. In addition to a number of seminars, conferences, she has over 150 publications. Madam has been guide to the Masters of Pharmacy, doctoral research, and goes on. And the list is endless. I welcome you all, ma'am. Welcome you, ma'am, on behalf of National Institute of Unani Medicine, Bangalore. Over to you, ma'am. Professor Zeenat Iqbal.
Hello.
Assalamu alaikum, ma'am.
Walaikum assalam wa rahmatullahi wa barakatuh.
Audible hai, ma'am. Well, I
Audible
welcome
hai,
you on
and
behalf of National Institute of Unani Medicine, ma'am, for the webinar.
thank you so much. Thank you so much. But I'm facing a trouble here that I'm not able to connect with from the laptop. So I'll just have to look into that for a bit. And the connection in the mobile is not that good. This is something which is unforeseen. I'm extremely sorry. Dr. Arshia, you just give me a couple of minutes? I'm trying again to join from the laptop.
No issues, ma'am. Take your time.
Okay,
No
please.
issues.
I'm extremely
What you
sorry
can
for
do,
this.
you just log out and maybe there are 2-3 windows of Zooms are open. Sometimes
Maybe that would be.
you just
I'll
log
just try
out.
to do that. I'll try to do that. I don't know,
Okay, no
maybe some
issues.
firewall message is coming in the laptop, which was never happened earlier.
No
Okay.
issues.
I'll just
Just
try
log out
to
and
do
log in
that.
again.
Surely,
Yeah,
surely. That's
okay.
what I'm doing from the laptop. Let me just try it again, inshallah.
No issues. You take your time.
Okay, okay. Dr. Amy has not been able to join?
She joined, but there was some technical issues. The voice was
Oh
not
my
clear.
God, the same. I don't know what is the issue in the laptop or the connection through the
Otherwise, you
laptop.
share your PPT. You can
That's the other
continue.
thing which I can do perhaps.
You just share the PPT. We'll run from here.
I've just called a technical person. So if
Okay.
that is okay, then fine. Otherwise, I'll do that only. Okay. Dr. Arshia, have you received the mail?
Just I will say. No, madam, I have not received it. Resend it. Resend it, madam. You can WhatsApp me. Madam, you can WhatsApp me. If it is large, it will not attach to it. Email.
Ma'am, did you receive it now?
No, ma'am. WhatsApp me, madam. No issues. Can you WhatsApp me?
Okay.
Yeah, I received it. Okay.
Ma'am, you can share the screen, Arshia ma'am.
Yes. Give me 2.
I trust it has opened up.
Yeah. 2 minutes.
Okay, fine. You take your time, no worries.
You can see the screen?
Not yet. You will be helping me out with this change?
Yeah, of course.
Okay, fair enough. No worries.
It's coming?
Yes.
The
It's
slides are
coming now,
moving?
Alhamdulillah. Alhamdulillah.
Okay.
The next one. That's it.
Dr. Zeenat, you can start your presentation.
Inshallah, I think I'll have to manage actually, I'm connected through the phone. Inshallah, it continues to work. The signal is an issue here.
Madam, you're audible. You can start.
Okay.
Okay.
Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. And a very warm good afternoon on a very cold Delhi afternoon this side. And I trust Bangalore would be a little bit more balmy. Absolutely apologies from my end as well for this technical glitch, but Allah has been kind to us and I think we are able to connect now. I trust I'm pretty audible. If somebody could indicate that to me. Dr. Arshi, I'm audible?
Yeah, you're very clear, madam. You can continue.
Thank you so much. The topic which was, I think at the outset, it's my humble duty to appreciate the efforts of Dr. Arshia, the chairperson of this seminar, and the efforts which she has made. It was during a conference that we had met, and Alhamdulillah, during those deliberations, it was discussed that we are going to have an international seminar. And I have an expertise in nano formulations. So it is something which is modern and something which the system needs to be introduced to the other pathies as well. So I took this opportunity and I'm extremely thankful to her that she allowed me to do so. And here I am with the presentation topic by the name of Onco Nanomedicines. I must just clarify, when I use the word onco nanomedicine, we are heavily borrowing it from the word oncology and then marrying it successfully with the word nanomedicines. And then we have tried to, I just took permission from Dr. Arshia that I would be maybe just tweaking my topic a little bit more. So I have taken up this reproductive cancers as per se, although as we proceed in the presentation, we'll come to know that most of the cancers will be in the category of solid tumors, and it would be wonderfully sort of connected with even the reproductive cancers. May we go to the next slide, which primarily is the content slide. We may move to the next one. So I'm happy that I'm connecting with all the sort of community, which is already a doctor's community and a tabib's community. And I need not emphasize much on the definition. Nonetheless, WHO has very clearly identified this as a disease which shows and characterizes with an uncontrolled growth of abnormal cells and which primarily invade the nearby tissue and lead to metastasis. And the incidence and mortality is a huge thing, a problem for not only the researchers, but for the patients and the treating doctors as well. So the next slide. Dr. Arshia, the next slide, please. The next one. So I think these are some of the pictures which are commonly in our memory. Then most of them are celebrities, so-called celebrities who have been in the public domain. And the common link between all these pictures is that they have suffered from the big C or the cancer, and they have been some of the, for example, Jade Goody, we lost her, but the others have recuperated, and most of them heavily relied on the nanomedicines. We move to the next one, Dr. Arshia. Another celebrity, and the reason why these celebrity pictures become important for us to relate is that unfortunately the common person or the common people, they are themselves not knowing what the disease is. But when these diseases are being suffered by the celebrities, it comes to a public domain and it attracts a lot of attention. So it is good in a manner that the celebrities, when we come to know that this disease is non-discriminatory and cancer has no discrimination, it surpasses all the boundary walls and it goes to all the economic strata, social stratas, and unfortunately, it is a one big commoner between all the stratas. We move to the next one, just to set the pace. So it is not limited to celebrities, all the common women and all the common people suffer as well. So cancer is a big sort of a commoner, one thing which is common in all the stratas and social. So it is a problem for all the communities and all the social stratas. Next one is a slide which primarily, there will be certain slides which I can skip and maybe I will not spend much time. Not to mention that when we talk of the reproductive cancer, it can be an ovarian cancer, a cervix cancer, uterus cancer. And because we are talking women-centric, so breast cancer is also included. Next one. As I just mentioned that there will be, the reproductive cancers will not limit themselves to only a few organs. They are not a single organ which is being left out when it comes to the causing of the disease. So there will be a lot of issues with the reproductive cancers, and the problems can be many and the reasons could be many. So there could be an exposure to the carcinogenic agents, or there could be a sort of a back role of hormone replacement therapy. There could be an exposure to the X-ray radiation or gamma radiations, and for that matter, infection with viruses, HIV or HPV. There could be many, many reasons by which these cancers come onto forte. We move to the next slide. The next couple of slides is talking about the statistics. So we have got the incidences just to make aware that how big this problem is. And there's a lot of mortality. You can see the blue bar, which is indicating the huge amount of mortality amongst the people who are suffering. And it is across the region, across the social strata, as I just mentioned. The next slide is again talking about the prevalence, and you can see that it is a clear and a present danger. We cannot ignore it, and all the varieties of the cancers have come up, and there's a lot of statistics which have been generated, not limited to India, but in the globe across. So the problem is huge and the solutions are to be sought after. We move to the next slide, and we again talk about the next two slides are simply a summation of the risk factors. So there will be certain common risk factors for all these 4-5 kinds of cancers, but there will be certain very specific one as well. For example, PCOS comes up a big risk factor in case of the endometrial cancer. However, the HPV virus is one of the biggest risk factors for the cervix cancer. It's just a summation. I think most of you would relate to this slide very well, being from the field. The next slide emphasizes again the risk factors which are primarily associated with the ovarian cancer as well as the breast cancer. In a nutshell, what we can say is that there is a huge variety of risk factors, which primarily most of them could be modifiable and some of them are non-modifiable if they are genetic predisposition. So we primarily have a huge plethora of risk factors which needs to be taken care of when we talk about these cancers. We move further. In the next slide is again, I'm sure that being clinicians, you will be very easily able to relate to this slide, that there's a lot of clinical manifestations. Some are very obvious, some are not obvious. So it is always the responsibility of a clinician to be aware that what could be the associations of these particular cancers. The more we are inclined to understanding these manifestations, the better it is for the diagnosis and for the patient further treatment. The next slide is primarily a connector for all these things. As we have been talking and progressing in this presentation, we now can be able to appreciate that cancers are primarily, if we talk about the cancers of reproductive tract and others as well, so if they holistically, we can call them that they are all solid tumors. So if we understand the solid tumors in general, and then maybe apply that information and knowledge to the reproductive cancers, we'll be in a better position to treat and diagnose them. The next slide is again a sort of reiteration of how a solid tumor looks like, and both digitally and pictorially, and the real sense that how the solid tumors comes out. The cancers and solid tumors, and primarily just a summation of whatever we have just spoken. So mostly when we talk of the solid tumors, they can be, mostly they are malignant, and they can be either sarcomas, carcinomas, or lymphomas. And also the main sites which are there besides the reproductive tract of women, they can be also present as lung cancer, prostate and colon cancer. And the tumors can primarily be benign or malignant, but we'd be more interested to talk about the ones which are the cancer metastatic ones, or which are malignant kind. So being a doctor, I think this is again very understandable that there would be different therapeutic modalities. And these therapeutic modalities can be understood as the invasive techniques. The one we talk about, the most radical type of therapy would be that of the invasive techniques, which would be in and around the various types of surgeries. The others could be the chemotherapeutics, which would be of more interest to us today as we progress in the presentation. There would be a plethora of cytotoxic drugs. There could be hormonal therapy. The latest one are your biological response modifiers, signal transduction inhibitors, and monoclonal antibodies. The miscellaneous would include the radiotherapy, where we usually use an external beam for radiations, which is again usually done in conjunction with the chemotherapeutic agent in order to get the best of the outcomes for the patient. Not to mention that there would also be a lot of progress in immunotherapy, targeted therapy, and gene therapy. As we progress forward, this slide is something very interesting to us as a pharmaceutical scientist, but I'm sure it would be equally interesting to all the practitioners and clinicians like you. When we talk of any drug, the drug doesn't descend from the heaven. It doesn't come from anywhere else, but from the laboratory researches, which is a very protracted and a long research of close to a decade and a half. We start from discovering the drugs which are there in our old documents. So when we start with the drug discovery, I would want your attention on this whole path of the drug discovery and development. We start using the first, we actually enter into the first part, that is your drug discovery. And usually these drugs which we are actually going to investigate are available in our public domain. When I say public domain, we usually have a plethora of literature, which could be Unani literature or Ayurvedic Charak Samhita, or for that matter, any documented thing, including our Holy Quran, where you would have a lot of discussions and a lot of mentions of the various medicines. So the first set of modern medicine discovery also comes from these data, which is documented well nowadays. And we start our journey with as big as 5,000 or 10,000 compounds. We have an inclination that some of the molecules which will be effective and they are being having that what we call as capacity to treat a disease. So we take that compounds and we do the chemistry about it. We do this elucidation of the structure. We do all the modern techniques to find out what exactly that chemistry behind that is. And once we are sure that out of that screening which we do, we usually use the word high throughput screening. So we have different types of screening which we do, and we find out that out of those 5,000, 10,000 molecules which we have started our journey with, only 250 molecules or little less than that are having some potential. So then these are submitted to the animal studies. So out of that animal studies, both on rodents and non-rodents, we primarily look forward to that whether the drug is actually anti-cancer or not, whether it is having some effect on tumor or not. We do a lot of studies on a lot of animals. And finally, after the screening, imagine that from 250, we are left only with 5 number of molecules. And this comes, and here comes the most important journey for this drug, where this drug is going to be submitted to the human trials, which would be around 3 phases, followed by a 4th phase of post-marketing surveillance, where the drug would be tested in healthy volunteers, followed by diseased people in phase 1, phase 2, multicentric. And after a lot of effort, a lot of hard work, we come out with one drug, and that drug is very, very precious to us. So I was just wanting to share this slide with a single intention that when we talk of the drug discovery or the molecules, the journey is a huge one and it takes a lot of time, billions of dollars, and then only we get that one pharmacological agent, which is your cytotoxic drug, or for that matter, a monoclonal antibody or any other category as well. So the next slide perhaps is giving you a little bit of snapshot of what are the various types of modern medicines which are available. You can see the names of these drugs. But these drugs usually will come with a lot of side effects. They would primarily have a lot of issues, even if they are good to go for the cancer treatment. And cancer in itself, as doctors, you must be very aware in itself, they will have a lot of sort of challenges, and then overcoming those challenges becomes a big, huge task. So the next slide perhaps is talking about some of the challenges of this current therapeutics. The cancer therapy is in itself is economically a very draining disease. So you know that when we talk of cancer patients and the treatment of cancer, one thing which troubles us a lot is the money. From where will the money come? The cost and availability of a drug is a big issue. The tumor heterogeneity is again issue. All the tumors are not same. So we do not have the same solution for the variety of the tumors which we want to treat. Then the drug resistance will be there. Then the patient response is sometimes not there. So it's not necessary that all the patients will respond in the similar manner. Sometimes despite our best of the efforts, we are not able to give the adequate amount of dose. So inadequate dosing would be a problem. Limited therapeutic window would be a problem. And lot of reasons of treatment failures can be there, and the disease progression goes on. And despite our best of the efforts, best of the medicines available, we still have a challenge to treat this disease. The next slide is actually helping us to understand that yes, the solid tumors generically will have all the problems which we discussed, but a reproductive cancer on the more will have a little more problems associated. So there can be an issue of a gonorrhotoxicity. There could be some associated neurotoxicity. And there is another big problem with women, which is coming up, or for that matter, males as well, which is referred to as your onco-infertility. This onco-infertility thing has given rise to a huge field in itself, that when nowadays we have onco-fertility doctors who are prescribing medicines while they are in the team of the doctors who treat the cancer. So this has come up in a big way because it is understandable that these chemical entities will definitely impact the fertility as well. And when we talk about the other cases, which are the other, I'm still on the slide here. And still on the last slide, that's it. So there will be a lot of issues which could be like associated particularly with the reproductive cancer treatment, which could include the impaired sexual dysfunction, then impaired increased risk of infertility, low efficacy of to reach the epithelial surface for of female reproductive parts, and there could be a lack of early diagnostic strategies. So needless to mention that although the solid tumors will be a problem, but the reproductive tract tumors are a bigger problem. So we really need to find out what could be the solution. The next slide perhaps is a solution I think, a slide of hope. So this is a recent slide which was primarily being floated on the different newspapers. And there you will see a common word throughout this slide, that is nano drug, nanotechnology, nano drug kills aggressive breast cancer, and so on and so forth. So there has been a lot of interest in developing solutions. And one of the key solutions which has come up is construction of the various drugs which can be nanotechnology potentiated or nano-based drug delivery system. So we move to the next slide, which again is a very recent slide, which I took from particularly, and you would be amazed to know that this is also a part of the syllabus of UPSC aspirants. So that is the level of the attention which nanotechnology is getting, and the type of sort of answers and the type of solutions which nanotechnology is offering is very heartening and very encouraging. So the next slide perhaps is going to help us understand that how this birth of nanomedicine happens. Honestly speaking, when we talk of nanomedicines, nanotechnology per se is a physics-based entity. It has nothing to do with biology or science of life sciences, which pharmacy people and doctors study. It is a pure physics. So it was I would say mid 80s, that it came up, and this scientist by the name of Richard Feynman, in one of his very important seminars, he declared that there is plenty of room at the bottom, and there can be a lot of usage of nanotechnology, and particularly in treatments and therapies. So these scientists, I will not take name of each one of them. These are some of the major contributors to the science of nanotechnology. And this nanotechnology, it's successfully married with the field of medicine, and something new, and the birth of nanomedicine came up. So when we talk of nanomedicine, more or less, the things which are, so this next slide, please. Nano, as the word suggests, is something which is small. It is dwarf. So the whole concept of this nanotechnology is based on the size. If this size would not have been there, it would not have been possible to do what we are aiming to do today. So this graphical presentation helps you understand that how small a nano is, or how small a nano system could be. It is 1 billionth, no zero hamare lagte hain. It is 1 billionth part of a meter. And you can appreciate on this slide in front of you, where you can compare a tennis ball with a molecule of water, which is 10 to the power minus 10, and the minus 10. And you can see that up to some level, up to the level of the period, up to the 10 to the power 4 size, we are able to say from the visible, we can see by use of our naked eye. And beyond that size, we are not even able to see. So we are playing in a field where you are not able to see the nanoparticles. We are playing in a field where the size of bacteria and the viruses are. We are playing in a field where DNA is attacked by a nanoparticle. So we are actually talking of certain entities which are very, very small. And the nanomaterials, like your nanotubes and nanoparticles, can be as small as 10 nanometers, which is much smaller than the bacteria itself, or the virus for that matter. So that is the beauty of nano, and that is why we say small is beautiful. The next slide perhaps take you a little more into the details, that why exactly we need nanocarriers. Nanocarriers primarily are easy to be used for tumors, because they can actually enter into the tumor, and they can be retained there for a long duration of time, which we refer to as an enhanced penetration and retention. And there is a lot of active mechanism which is involved in taking the nanoparticles into the tumor milieu. The next slide, and
Ma'am, you are not audible.
Ma'am, voice is
Maybe some network issue. Madam will be joining shortly.
We are sorry to the delegates, as there is some network issue in her place. She's joining. Please be join with us. She's rejoining. The speaker is rejoining. Please be rejoin with us. Okay? Don't leave. She's rejoining us.
Audible?
You're very clear, ma'am. You can join.
Okay, fine. Thank you so much. I think inshallah I'll start again. Just shut this door, beta. So I think I was just talking about the use of nanotechnology, and I think, thank you so much, Dr. Arshi. I'm extremely sorry. I don't know why this issue is happening today. So this slide is again a very encouraging slide, which is helping us to understand that right from
होंगे वहाँ भी, तो उसके लिए भी रुके हैं.
उनकी बीवी गाजियाबाद में है ना, वो वहाँ जॉब करती है. Could you please mute your devices? That would help me.
साथ होते थोड़ा बहुत दिमाग.
Thank you so much. Thank you so much. So I'll just recheck if I'm audible to all of you. Somebody indicate that to me. I trust I'm audible. So I was just talking about this timeline. This is again a very encouraging slide, which is helping us to understand that right from 1964 onwards, there have been a lot of things and a lot of work being done in the field of nanomedicine, and particularly to treat cancer. So we were able to talk about the liposomes. We had the discovery of the small interfering RNA and many, many more. We move to the next slide, and perhaps that will be a very basic understanding for all of you. So when we talk of the nanoformulation, the big question comes that what exactly is nanoformulation and how it is being different from a normal naked drug. So mostly as doctors, I think you must be prescribing a lot of tablets, capsules, and for that matter, injectables. And one common thing which usually you see is that you imagine and you understand that the drug would go into the GI tract if it is an oral formulation, and primarily it will break into small particles, and finally it would be sort of dissolved and then finally reach the bloodstream, and that drug would then act there, and finally, inshallah, you get the outcome or the therapeutic outcome. What exactly we do in nanoformulations is that we are not leaving the drug naked. what we do basically, we take the drug and the API and we take a lot of excipients, a lot of procedures will be there, and we make a particle which is now embedding the API. So it is basically a clothed drug. We are actually mantling the drug and putting some clothing onto that drug, and then the drug is not naked, it doesn't enter into the system naked, it is having a carrier onto which it is loaded. So that is the basic concept. It would be a little more complicated as we move further, so but this basic understanding will help all of us. So the advantages which will be given by these types of nano formulation construction would be, it would result in a decreased drug resistance. It will decrease the toxicity because it is not directly interacting with the tissues. It will enhance the oral bioavailability, enhanced rate of dissolution, solubility will be increased, drug targeting ability is increasing. So it will reach to the target more easily than to remain in the non-target sites. And because of this, there will be a huge reduction in the side effects. There are some of the things which is important for us to know as we progress into the presentation. The next slide is again giving you an insight. This slide is again maybe the words would be a little not very familiar to all of you, but these are the words which if you take a research paper or if you just Google out the various types of nano formulations, so there can be a variety of them. Like we have the different conventional formulations, we have liquid dosage forms, we have semi-solid dosage forms, we have solid dosage forms, parenterals, whatever kind. So the same thing if we extrapolate to the pharmaceutical formulations of nano-based, there can be a variety of them, and primarily they are polymer based, they can be limit based, and they can be non-polymeric, and sometimes they are nanocrystalline formulations. So mostly what we usually do, we are mostly doing the polymeric based drug delivery system and the lipid based drug delivery system. There will be a variety of them. These names like nanoparticles, micelles, drug conjugates are very technically used depending on what type of method has been adopted or what type of delivery system have been designed. So we move to the next slide, and I think this helps us understand a little more in a bit detail about these nano systems. We have the polymeric one. As the name itself suggests, that we will use a biodegradable and a biocompatible polymer, which would then be used in a manner to encapsulate the drug which is of interest to us. So we have a polymeric membrane into which the drug is being loaded, and this inner core consists of the drug, and then we make it into a particle form between 10 to 1000 nanometers, and primarily we are able to create a multiple system which is having different particle size, particles in that. We also have these dendrimers, and some of the categories can be your micelle formulation. I would like you to appreciate one thing, that the drug which is being shown in the core, or the drug which is shown in the middle of this micelle, this drug is normally we work on synthetic molecules, but we have also tried our hand to use this carrier system to deliver drugs of plant origin. So we can have a drug like from Andrographis paniculata, from Calmake like andrographolide. We can have a drug like thymoquinone, which is obtained from the black seed, and we can have anything for that matter, and which can be encapsulated and can be delivered like we deliver the synthetic molecules. We move to the next slide and maybe a little bit more insight into the variety of lipid based systems. So you must have heard of these. A lot of cosmetics nowadays are lipid based. Just to give you a little bit of feel of what this lipid based system is. So you would be having anti-aging cream, which is having a solid lipid nanoparticle. So the idea basically is that lipids are usually inherently available in the human body. So they will be more acceptable. They will be like more acceptable by the human system. So the formulations the formulation scientists are harnessing the potential of those lipids and then using them in the form of the carrier system. So we use different types of lipids to encapsulate that drug in different concentrations. So we can create solid lipid nanoparticles. If more of solid lipid is used, the name solid lipid comes. If more of your liquid lipid is used, we can use the word liposomes for that matter. And a lot of liposomal drug delivery. Maybe you remember the COVID-19 days and this formulation amphotericin B that was primarily having a lot of liposomal drug delivery, and these liposomes are having a very good advantage that they are hydrophobic in nature and they would be entering into this antifungal much easier. Similarly, they have got a beautiful structure which you can see on your slides, which can be then manipulated to deliver different types of drug, whether your drug is water soluble or water insoluble. Again, I would like to reiterate here that the selection of the drug is not limited to a synthetic molecule. We can definitely go in for a molecule which is a herb origin or a plant origin or a phytochemical for that matter. We just have to tailor and customize the properties of that drug, whether it is hydrophilic or hydrophobic. And if we are able to do it, we can definitely load any kind of drug into liposomes. The other system on the extreme of this slide is that of nanoemulsion, which primarily is one of the very commonest of it. You all have been using emulsions and making emulsions where you use a water phase and an oil phase. These two phases, when they are brought together with the help of a surfactant and crushed properly, we get very globules, which is biphasic in nature. If we do it in a manner that the particle size of this emulsion is a nano range, this emulsion will now be called as a nano emulsion. Again, a very suitable preparation for delivering highly active dermatological products precisely. You will get a lot of nano emulsions of anti-aging creams, and you definitely can also use it for cancer treatment, as I will just explain in a bit. So we move further and we talk about another set of, I'm sure that these are little technical things, but nowadays science is borderless. And when you as clinicians and doctors, you get a formula in your hand, somebody tells you that this is a neosome of methotrexate or an anti-cancer drug, then you should get a small little picture in your head that how these neosomes will be. And let me remind you that all these neosomes and artificial exosomes, or for that matter, liposomes, which we just mentioned a bit back, all of them are not visible to our eye. The pictures which you are showing have been taken from scanning electron microscopy and the transmission electron microscope. And this is a simple arrangement of the lipids and all. You can just appreciate in the case of neosomes that you have got a hydrophilic head and a hydrophobic tail. Then you are having an aqueous compartment in between. And you can definitely assume and think that the center of this whole neosome can very easily encapsulate a water-soluble drug. So if you have a water-soluble drug, you would choose neosomes. If you have water-insoluble drug, you might go in for solid lipid nanoparticles, or for that matter, liposomes. Similarly, there can be an artificial exosomes, which are again prepared by lipid bilayers, very similar to your liposomes. And you can simply say that you can find products like paclitaxel encapsulated exosomes, which can be used for the treatment of pancreatic and can be for pancreatic cancer per se. We move to the next slide and just a little bit more about the other types of nanoparticles. You must be hearing a lot of news that there are silver nanoparticles, gold nanoparticles. They are used as an antibacterial thing. You will be amazed to know that the antibacterial deposition or the encapsulation is being used even for the refrigerators. You can have silver nanoparticles which are put into your socks. And when you wear these socks, the silver nanoparticles are loaded with, they themselves are antibacterial. They can also be loaded with some other antibacterial drug. And while you are wearing your socks, the drug will be released, and primarily the bad smell in your socks or in your feet, which usually troubles a lot of people, can be controlled. Similarly, we can have magnetic nanoparticles, silica-based nanoparticles. The idea to show these slides is just to give you a smattering or a little bit of picturization of that what various types of nanoparticles could be. This is something I think the next slide talks a little bit more about the quantum dots and the carbon nanotubes. These are the inorganic type of nanoparticles, and quantum dots, as clinicians, you must be aware that they are used for more for the diagnosis purpose. And the reason why they are very suitable is that they can accumulate in tumor tissues. The tumor visualization can be there, and it is a non-invasive diagnosis. So there has been cases where the HER2 positive tumors have been detected and diagnosed by virtue of using the quantum dots. Similarly, you must have also heard about the carbon nanotubes. They are again little rod shaped, about 20 to 1000 nanometers, can take up the drugs, and it has been found that the drugs like methotrexate, when loaded into nanotube, have significantly increased the anti-tumor action. We move to the next slide, and this is the next slide, please. So all these things can be referred to as a nano tools, or it is very understandable. The better the tools are, the more sharpened they are, the more I would say pinpointed they are, the better would be the outcomes. And nanomedicines per se, they have been now very routinely used in clinical practices. Some of the examples are in front of you, where we are actually using the nanoliposomes and the antibody and the conjugates made by the company Celltech, and we have the molecule polymer drugs and conjugates by the name of Duncan. So the idea of this slide is to just reiterate that nanomedicine is no more a dream, but it is an actuality and a reality where it is being used by clinicians very, very normally nowadays. For the cancers, we have to be a little more I would say specific, and little more, I would say, strict in designing, because our tumor cell will primarily have more complications and it will have more challenges. In the next few slides, I'll talk about that what are the actual challenges which is there in the cancer treatment, and as doctors and clinicians in making, you would understand it much better than others. So you can just appreciate that the tumor cells can be treated, or the cancer can be treated by using a large variety of drug delivery apparatuses, which could be your tumor can be having a passive targeting and active targeting. You can use for imaging and cancer treatment. You can use the nanoparticles for diagnosis and treatment together, which can be called as theranostics. You can have inorganic nanoparticle, you can have lipid, polymeric, and whatever kind of particles in order to be used as advanced nanotools for cancer. Needless to mention that when we try to treat cancer with a conventional drug delivery system, we are very much aware of the fact that the drug leads to a lot of side effects, which sometimes makes the patient compliance a very poor thing. So this is something which is very interesting to be noting, that as we progress in making more nano formulations, they are more suitable for the treatment of the biggest problem called cancer. So we move further in the next slide, and this is a simple. The next slide is, it is actually a paper which we had written long back. And the paper had this, the genesis of this paper, and why did we actually wanted to write on this? It's a very negative paper, not in favor of nano drug delivery, but we wanted to know that what exactly the reasons, because I think again, you must be aware, being from the field, alhamdulillah, you people must be very aware that despite so many efforts of nanomedicines, nano formulations, the translation rate of success is very low. There's a lot of discoveries, the drugs are there, but you would still see as doctors pathetic condition that the treatments are still not very, very effective. So there could be certain reasons why this nano drug delivery system are usually not very much helped by these nano systems, or this cancers usually pave a lot of challenges. So we tried to write this paper, and the reason we in this paper, when we reviewed it, and I think it's a very, anybody who's interested in cancer would really love this paper, I'm sure. So usually there was this, we tried to do a comparative data of nano delivery system, and we tried to find out the reasons. And one of the major reasons was that we were not able to follow the in vivo tracing was not there. The targeting was a problem. The dosing was a problem. Sometimes may I request you to please kindly mute yourself? Thank you so much. So when we were talking about this, so I was just mentioning that there was a lot of issues with nanosystems. So it is like always very heartening to see that nanomedicines are there. Now everything will be hunky-dory, everything will be good, and the cancers will be treated. But the outcomes are still not very heartening. The outcomes are still not very encouraging. So the next few slides are, because mashallah, you all are from the field, and so also us, that as clinicians, we should understand that why there could be a failure. So this failure would help us to understand that there are certain reasons why these nanoparticles could fail. And I always say one thing to my students as well, that if we are able to understand the disease more, we are able to actually understand to give a better solution. If I know that this will be a problem in pathophysiology of my disease, I will make my formulation accordingly. So one thing is very clear, that nanoparticles will definitely come into contact of various physiological conditions. In the normal physiology, it's a challenge, the type of blood flow, the type of tissues, then opening up of the formulation, then reaching to the target is a big issue, because the body is not going to let it easy. It will not allow it as if it is not having any barriers. So body is made like that. It will give a lot of barriers, which are biological and physical barriers. So, and in case of disease, the barriers become all the more difficult, because now the pathophysiology has changed, the vasculature has changed, the interaction of the system with the blood vessels will not be that easy as compared to in a normal tissue. So normal may it was so much difficult. So we can understand pathophysiologically disturbed tissue will be more challenging for that nanosystem. Let us move to the next slide, where we will be talking about the first thing or the first challenge which the formulation or any nanosystem will actually come into contact with. I have been telling you in the beginning itself, I trust you remember and you were there. I'm extremely sorry for a lot of disconnect in between, but I'm sure that you must be remembering my one statement that when we talk small is beautiful. So the whole idea of nanosystem is that they are too small, and while they are small, they're actually trying to get into the biological system and reach that particular site and remain stable during that journey of reaching to that particular site, and then give that outcome which we desire from it. But unfortunately, that is not the reality. The system will come in contact with macrophages. The system will be like sort of, it is going to be a foreign entity. It will primarily be captured by the macrophages and all. So the formulation scientist does one thing very nicely, that in order to make the reticuloendothelial system and all the other immune system foolish, so in order to fool that system, what we do certain magicry. We do something, we call the word, it is a pharmaceutical magic. But exactly what we are doing, we are taking that nanosystem and we are covering that or coating it with certain other polymer, which will not allow its recognition. So one of that polymer is called as your PEG or polyethylene glycol. So we cover that, and we usually allow that covered particle to reach, and that will be camouflaging its effect, and it will not be captured by the cell's defense mechanism. So that is one way that we make sure that the circulation is very good. Because if the circulation is not long, just imagine that if a nanoparticle enters the system, it leaves, where how will it affect? So it has to take the huge journey. Imagine that you are giving an injectable. So from the injectable, it will get into the intravenous route, and then it will go through the whole gamut of the journey in the human body, and finally reach the breast tumor, or for that matter, the cervix tumor. So any medicine takes a huge journey in the body itself. So if the nanoparticles are not safe or not stable during that particular journey, then the whole purpose is defeated. So pharmaceutically speaking, we use a lot of systems, lot of mechanisms by which we camouflage them and we make them reach to that particular site of action. So another option by which we can do is that the next slide, please. So one slide, next slide, please. So one we were talking about was the addition of the pegylation or the last one, please. So I was just trying to reiterate. No. That's it. Thank you. So when we're talking about this camouflaging, so one of the ways is also to camouflage by using the platelet membrane cloaking. So we can use even the platelet membrane. So this is again a very interesting thing, that we take out the platelets, or we take out even the WBC, and then we use those cloaks or these outer covering of these cells in order to deliver our nano formulation. It is very understandable that because it would be human in nature, so the rejection chances are very less, and the stealthiness or the movement of that nanoparticle in the body system will be much, much higher. We move to the next slide, which is again just giving you an insight about the various available mononuclear phagocytic system. So this just gives you a little bit of idea. I need not emphasize. I simply reiterate the fact that the body is ready to capture the nanoparticles, but we as pharmaceutical scientists make the nanoparticles cover up and they will reach the targeted site. The next slide is again giving you a little bit of idea that how this, I was talking about that how these nanoparticles will be carrying forward. The next few slides, I want your attention on the next few slides with the simple intention that what exactly is happening in a biological system. Just simply making it, giving it to a patient is one thing, but knowing that how it acts will actually add on value and will help us to do better research on that. So I just want to appreciate, and I trust this diagram is very clear and much clearer to you people, that whenever we talk about, you can see the blood vessels and you can see the nanoparticles. And these nanoparticles, by virtue of, and it is again understandable, that in case of a cancer shell, cancer tissue, the amount of the barriers will be less stringent. The barriers are usually broken up and there will be a process of intravasation and extravasation. So the nanoparticle system per se takes advantage of it, that based on the size, if the particle size of the nanoparticle is small enough to get through that blood vessels, so you can just appreciate this green colored dots, which are the nanosystem and which are getting extravasated and entering into the metastatic tumor because the vasculature here is disrupted. So if I make my nanoparticles small enough to get into 3 leakage points, then that particle will be easily reaching to the tumor site without much effort. So the next slide, please, is the next slide is again the sort of extrapolation of the thought which we were just discussing. Is you can just appreciate in the diagram where you can see the pegylated nanocarriers. The last one, the previous one. That's it. Thank you. So when we are talking about, I just want to make this one small sort of submission here, that you can see that pegylated nanocarriers, they're actually getting into this milieu of this tissue, that is extracellular matrix tissue and the tumor tissue, just because of this extravasation and the vascular burst which is happening. And in case of the other category, what we can do, we can actually put some ligand facilitated extravasation. You can simply imagine that if the nanoparticles is being received by a receptor, that reception will be much more dependable. So we can have a nanoparticle which is by default entering into the tumor because it is extravasating through the disrupted vasculature, and in the other case it can also hinge on to certain receptors and enter into the tumor milieu. The next slide, please, is again a slide which is, I'm sure that this primarily is going to really bring back to your memory as doctors and all. I think this must be a part of your syllabus where you understand that how a tumor looks and what are the microenvironmental barriers of that. You can appreciate and you can see that the tumor definitely is not normal. It is having certain very peculiar conditions which it undergoes. It will be having necrosis. That means it will have hypoxia. It will have an abnormal vasculature. It will have a high interstitial pressure. It will be having an aberrant increased stiffness, and it will be having not so smooth surface as well. An absence of lymphatic vessels is there. Exactly this micro environment. I read it and I understand it, and based on this, I make my drug delivery system. So if I am having a nanoparticle, I will create a nanoparticle which will work better in a hypoxic condition. So I'll give you some examples in the subsequent slides that how this works. So my only submission here in this, that if I understand the microenvironment or the disease the best, I will be able to offer a solution much, much better. So the more I understand the disease, the better outcomes I will be able to give in the formulation as a formulation scientist. So the next slide perhaps is just giving you, I think this makes a lot of sense to all the clinicians here. So how do we know that what type of tumors will be having that EPR effect? I will repeat, this EPR is a highly reliable fact that because of the poor vasculature and because of certain disruptions in the pathophysiological condition of the tumor, it allows the entry of the particles, and by default there is a collection of those nanoparticles into the tumor tissue in which the drug will be delivered. So you can see that there are certain tests which can be done. So there will be certain, they use the word hot tumors and cold tumors, which is just simply to just make you understand that if the sort of tumor has grown well and it is circulated well with the blood vessels, the accumulation of the nanoparticles will be much higher. If the poorly functional vessels are there, the EPR will be medium. And if there is a lot of desmoplasia and high interstitial pressure, fluid pressure, then it will be a little bit of negative for the accumulation of the nanoparticles. So mind you, the reason why this has been told you is that based on the type of tumor, if I'm able to understand what type of tumor phenotype is, what type of tumor behavior is, I can actually decide whether I will make a polymeric nanoparticle or whether I will go in for a gold nanoparticle or whether I should go in for a lipid-based nanoparticle. So the more I understand my disease and the tumor situation, the better choice I can, because I've shown you a lot of types of nanoparticles. There should be a question in your mind that which one is suitable for what, which type of tumor, because tumors are not same. One glove doesn't feel all, is not the same for all the hands. So we have to change the gloves. So we have to change the type of nanoparticle as well. So let's take it forward and just this understanding. So this is just a summation. The next slide only. The next slide talks about the enhanced permeation, which I've just explained to you. We move to this. So not only this microenvironment, but at the level of the, this one, the previous one, please. Yes, thank you. So I was just mentioning that once the cell, you can see that the nanoparticle has somehow entered into the blood, and from the blood you can see the blue particles, they are getting into the tumor by the virtue of the enhanced permeability effect. Not only this is sufficient, now this nanoparticle have to overcome the intracellular barriers. You can see the other part of the slide, which is telling us that one of the commonest way of engulfing the nanoparticle is by the natural process of endocytosis. You can appreciate the early endosomes formation, then the endosomal escape, then delivery of the drug, even to the nucleus. So when you talk about the cancer drugs, some of drugs will be intercalating agent, some drugs will work through the cytoplasm, some work will go and maybe intercalate with the topoisomerase, some will do this, some will do that. So you must also appreciate clearly in your heads that the nanoparticle is made in a manner that it should go inside the cell, release the drug there, and then the drug is available for its actual site of action, whatever way it works. So we move to the next slide, and this is again a reiteration and just helping us. Next one, please. This one only, thank you. So it is this one only, the last one only. So this is just giving you an insight into that. You can see that there's a lot of, you can appreciate this green and yellow colored particles, which are the nanoparticles. They are going through the blood tissue and entering and extravasating into the tumor. And there is a dense ECM, elevated IFP is there, solid stress is there. It needs to get deep penetration and then finally getting internalization. So we cannot simply limit ourselves to making a nanoparticle. We have to think and visualize that how it will enter through the tumor, how it will be taken up by the cells, how will it penetrate, how will it internalize, and so on and so forth. And luckily at every stage we have got now very advanced techniques by which we can actually see whether the cellular uptake is happening, and then we try to inshallah replicate into the human system. The next slide perhaps is again a reiteration of the same thing, that how the passive targeting is happening and the active targeting is happening. I think by now it must be clear. For the passive targeting, only the vasculature deficit is helping us out, whereas for the active targeting, there has to be receptor mediated recognition. So there is a concept of ligand based. So if we are able to understand that what protein is overexpressed in that particular tumor, then we can have primarily a complementary peptide onto the nanoparticle attached in the form of a ligand. So it will directly go. For example, there is a tumor which overexpresses transferrin. Then we can usually, and there will be transferrin receptors there. So we can make a nanoparticle which is decorated with transferrin, and that transferrin based decorated nanoparticle will actually land itself into that receptor, which will be then internalized into the tissue and then actively released and actively taken up. So normally we rely on the EPR effect, which is by virtue of this, I would say, this passive targeting, but we will be more happily able to talk about the fate of the nanoparticle if it is actively targeted. For that, we go deep down and we understand the concept of receptor and ligand. We move to the next slide, which is again a little bit a better diagram. You all know that there will be a lot of, I will just mentioning that there will be a lot of associated proteins like integrin, PSMA, CD44 for delivery. So if I'm able to understand this, you can just see that if I'm able to capture this essence that which is overexpressed at what tumor, I can use the same thing like transferrin and folate, which can be ligand based, and then I can target my drug better. The next slide perhaps is just again giving you a little bit of insight that we can have the ligand in the form of an antibody. You can have a small molecule as a ligand. You can use an aptamer as a ligand. You can use a carbohydrate even as a ligand, protein and a peptide. I have this one beautiful example. It is just crossing my mind that we know that when we are, where the cancer is always hungry, the cancer is wanting more and more food. So what we do is that we take this nanocarrier and we coat this nanocarrier with a carbohydrate like dextrose, sucrose, mannitol and all. So the cancer will be happily engulfing it because it's the source of energy. Mannitol is there, dextrose is there, and it does not know that inside deep within the nanocarrier, paclitaxel is sitting or topetacon is sitting. So the drug, the nanoparticle will be engulfed very happily by the tumor because it is having the food on the outer covering as a carbohydrate. But when it is engulfing it, it is not knowing that the energy will be given, but inside the belly of that nanoparticle, you are having the drug sitting there. So that actually is one of the beautiful ways by which we can befool the tumor and we can actually deliver our anti-cancer or cytotoxic agent. The next slide perhaps will talk a little bit more about the microenvironment based drug. We can move to the next slide. I think we have understood this well, that the microenvironment will be giving a lot of sort of challenges. So we can have two types of deliveries based on the microenvironment. If I know the microenvironment, my tumor, I can either have a non-stimuli responsive release, which is a simple passive drug delivery where I simply take a nanoparticle which is polymeric. By virtue of its particle size, it will reach the tumor. But I have an excellent way, the next slide, I have an excellent way where I can use the presence of certain specific conditions in the microenvironment of the tumor as the stimuli for my drug delivery. By now, we are aware that our tumor cell will be having different pH. It will have an acidic pH. It will have a high redox presence. The reactive oxygen species will be very high in concentration. The temperature of a tumor is more than the temperature of a normal cell. There will be very peculiar type of enzymes which are present there. Then it can also react to the light stimuli or photodynamic therapy stimuli. So based on this knowledge that there are certain peculiar conditions in the microenvironment, I can actually devise and manufacture very specific category of nanoparticles. This will be really very informative and very I would say encouraging to look at. So you can just see that we can either have an endogenous, next slide please, we can have an endogenous stimuli, which is inside the milieu of the tumor. We can have a pH change, we have a different enzyme, we have a different redox, and we can even use the outside stimuli like the magnetic temperature, light, etc., to actually stimulate the drug delivery. In the next few slides, I think inshallah I will be able to appreciate it more. And all of these nanosystems which we have now being known as liposomes, magnetic, they will be stimulated either endogenously or exogenously to deliver the drug, and they improve the anti-tumor therapy. So let's see the next slide. This next slide is primarily, I'm just giving a couple of examples that how the various types of conditions in the microenvironmental, environment of the tumor can be sort of harnessed for the treatment of that cancer. So just to begin with, I just mentioned that the microenvironment of the tumor will not have normal pH. It would be acidic in nature because of a high amount of lactic acid production and many other things which is happening there, metabolite and many things are happening there. So what we can do is that we take a simple, there's this polymer by the name of NIPAM. So we take this polymer which will be not melting at a normal body temperature of 37 degrees centigrade. But as soon as that nanoparticle reaches to the tumor, it is resulting in the melting of that polymer and delivery of that drug. So that is a very excellent way of delivering the drug. The whole system of the body, it is not doing anything, but as it reaches to the tumor, which is high in temperature, and sometimes the temperature could be as high as 41 degrees centigrades, and the simple presence of a high temperature, the acidic pH there. So the two things, I'm sorry, I just mixed up the two things together. So we'll just first talk about the acidic thing. So when we talk about the pH sensitive, so there will be very much the designing of the nanoparticle is done in a manner that it will only release the drug in that pH which is there in the tumor. And the pH in the tumor is acidic in nature, where the pH in the other parts of the body at that point of time are neutral or 7.4 or 6.8 around that. So I use this condition in the microenvironment of the tumor to actually deliver my drug. And this is going to make it targeted drug delivery. This will help me to only take the drug only at the site of action and making that system much more effective. We move to the next slide, which talks about the temperature-based delivery, the one which I was just mentioning. So this is again a beautiful idea where I know that the temperature of the tumor will be higher. So I select that type of polymer, I select that type of drug delivery system, which is going to be intact and stable and having integrity while rotating or revolving in the whole body system. But as soon as it reaches the temperature of the environment of the tumor, it is going to be melting out. It is going to react to those hyperthermic condition, and it will release the drug in a huge bolus at the site of the action. So simply selection of a temperature sensitive polymer is going to make my drug targeted to a tumor which is primarily having higher temperature. We move to the next example where I can have a system which is a ROS stimulated drug delivery. It has got two ways. Sometimes it is said, some of the researchers say that it is better to increase the ROS conditions or the reactive oxygen species condition that make it as high as possible. And once it is high, it is not going to support the growth of the cells. So either I can increase the ROS conditions by my delivery system, or else what I can do is that I can actually use antioxidants. I can use certain specific drugs. And what better than the antioxidants which we get from this source of black seed. And we are able to get thymoquinone, we are able to get quercetin, we can get so many phytomolecules which will be reducing the reactive oxygen species. And once I'm able to control the reactive oxygen species, I am actually making the cell more normal, and I'm actually ameliorating the cancer, and I'm actually making my drug delivery system effective. The next slide perhaps is would be of interest to you, that I will not limit this whole systems which we have just discussed are not limited to delivery of only the synthetic molecules. We can definitely take certain phytochemicals as well, nutraceuticals as well, which have got promising results as antioxidants and all. So we have this one typical example of the indoles which are found in cabbage, which are able to reduce the effects of estrogen, and they can be actually used for the treatment of or the development of the breast cancer. The next slide perhaps is again of interest to all of you, where primarily I've tried to just capture the various types of phytochemicals, beginning from curcumin, diadazine, ellagic acid, eugenol, which have been successfully encapsulated in different forms of delivery systems, be it a magnetic nanoparticle, PLGA nanoparticle, PEG-based nanoparticle, and it is giving the delivery to the breast cancer and the other thing and successfully treating that. So there's a lot of hope by which we can actually use the potential of nanotechnology to deliver drugs and formulations which are based on plant origin and which are having phytochemicals per se. We just the next slides you can move quickly. They are just some of the review papers which have a lot of sort of information. Anybody who's interested can go through this. You can just pass through the slides quickly. We have the integrating of the nanotechnology into cancer care. The next one, please. The next one, please. So somebody who is interested to know about the various methods can refer to this journal. Then emerging nanoparticulate system. There are some of the review papers. So we move to this slide. We can come to this one. Thank you so much. So usually after this, next one, please. So these papers were just a snapshot. They're easily available on the Google, and you can just find a lot of papers which will help you now after this lecture. I appreciate, inshallah, I hope that you will have some feeling about the nanoparticles and how they are synthesized, and reading these papers will reiterate your information. So ultimately, when we talk about, so ultimately any formulation is useless until and unless it has got clinical success. As doctors and practitioners, you would also be thinking that, okay, this formulation is not working. Why is it not working? So there has to be certain factors on which the nanoparticle delivery and the success depends. First of all, they should remain stable throughout the time of circulation. Number two, they should be able to overcome the physiological barriers and gain access into the anatomic side. Then only it will be effective. The bioavailability has to be high. The safety profile has to be acceptable, and there has to be a continuous research and how these characteristics of these nanoparticles can improve and how they can be used in the cancer treatment. So nanomedicine is a dynamic science which is continuously moving to keep on answering these questions, that how we can keep on changing the various, we understand the disease, we know what are the challenges, so we keep on making our formulation more certain to be delivering by changing the parameters, by changing the size, by changing the shape. The next few slides, primarily the next slide please, is again what exactly as scientists we are doing. So we have to actually overcome. By now, as clinicians and practitioners, you understand that how difficult would it be for any nanoparticle to reach to the site, but we try our best and we keep on doing some work on this, that how we make these nanosystems work. The next slide perhaps is a whole summary of what exactly we do. If we do our science, if we do our nanoscience, and if we make formulations, what are the things which we bear in our mind, and how do we start our research? So the first thing is that, yes, please. I trust I have some time. Can I continue?
Sure, ma'am.
Okay, please. Okay, fine. I think there was somebody saying something. It's okay. Thank you so much. So what I was trying to mention is that this slide is going to help us understand that what exactly is the crux of this whole research. The first thing is that we want the cancer. We should understand that cancer has to be targeted. We need to have some target clear in our mind. So we need to know the disease well. So we need to understand the pathophysiology. That is why I always believe that a real pharmaceutical research cannot happen without the help of a clinician. If we have a doctor in our team who understands the cancer better, for example, or disease better, for example, he can tell us that this is the situation and this is what needs to be treated. So we need to understand the underpinnings of a disease. The second thing after that would be that I have a lot of tools available. You must have heard of pharmacological networking. You must have heard of molecular docking. You must have talked about or heard about the druggable sites and computer-aided drug designing. So if I am a researcher who really want the bullseye to be hit and I want my success as my formulation, I need to take the best drug. Nowadays, a lot of interest is happening that people, because of the side effects of the synthetic molecules, they are trying to combine the various nano formulations with the phytochemicals, so to reduce the side effects. So if I know that my one drug, paclitaxel, is going to be cytotoxic, but my other drug like quercetin is going to support paclitaxel by reducing the ROS species and acting as an antioxidant. So the combination of these two will be a better one. I understand the pharmacology which is involved. The more I understand, the better it is. After this, when I understand all these things, I need to decide whether my formulation will go through an oral route or through a loco-regional route, or it will go through an injectable site, and so on and so forth. So I understand the limitations of nanoparticles. Then I put my energy. I look into the patient strategies. I look into the conditions of the tumor, and then I decide that what all things should be done. We move to the next slide. So it actually talks about whatever I have just said. It just simply puts that I will keep on developing approaches which will increase the tumor accumulation and penetration. I have to keep on expanding my toolbox. And one more thing which is very important, I am sure that as clinicians you understand it better than us. that we cannot have a similar type of nanoparticle system to treat any type of tumor. There would be an oral mucosa tumor or a squamous cancer in the mouth. It needs a different types of therapy. So I will not go in for nanoparticle. Maybe I will create a patch which will be like stuck onto that tissue. Maybe in case of a GIT tract, I know that there will be a variety of pH conditions there. My nanoparticle will not be able to remain stable. So I will do some camouflaging and I will maybe deliver my nanoparticle by some means directly to the colon so that it does not go through the vagaries of the pH in the stomach and doesn't get destabilized. So I will put some polymer which will only go and release the drug in the colon cancer. So these are some of the things which I will, and of course, there will be a continuous help from the clinicians who will keep on having the clinical trials and giving us feedback that what type of formulation is to be required. So there will be a lot of things have now happening. I think you must be aware that these techniques of ultrasound and hyperthermia and all can be very well used to, next slide please, can be very well used to deliver the drug in a better manner. Next slide please. That's exactly what I was trying to mention, that if I know the microenvironment better, I can have. So this slide, again, this will take a lot of time, but primarily I will just very quickly summarize that this is again a direction for us, that we understand, we stratify the patients. The concept of biomarkers have come up. Now, if you want to go in for, you suspect a person is suffering from prostate cancer, you ask him to go in for some blood test and you get biomarkers and you surely tell whether the person is having cancer or not. Similarly, there's a lot of other cancers which are associated with certain biomarkers. So this diagram primarily is just helping us to understand that the first thing we should do is that we should stratify the patients based on the biomarkers. Then we should go in for the various barriers which can be present there. And based on those barriers, we should treat whether we should start creating a formulation which will be able to overcome all these barriers. So this is, I always say that this is a beautiful slide which helps and gives us direction that how primarily we can actually look into, for example, if I want to go in for the treatment of a lung cancer, then definitely I will have to ensure that the moment the nanoparticles reaches a lung, number one, they cannot be too big, they cannot be too small. They have to be of a particular size. When they will be sucked in, they should have some capacity to get associated or attached with the mucin which is secreted in the lung. So that means it should be there in the lung epithelium and it should restrict there, retain there. So I will prepare a mucus penetrating nanoparticle for a lung cancer. Similarly, I will prepare a mucus penetrating nanoparticle for a cervix cancer where I know the lot of secretions are there and my nanoparticle will get adhered. So depending on what site of action is required and what type of drug, what type of sort of challenge the organ is giving us, I will have to. So this slide actually submitting, and not only that, I will primarily be looking into the immune system, that how I can surpass that, and I will also determine the pharmacokinetics of the disease, and finally I may take help of the genomic or CRISPR technologies as well. The next slide perhaps is again a little bit of... Yes, please.
Ma'am, we are running short of time, ma'am. First speaker also we need to continue.
Okay, I can, do I have 5 to 10 minutes? 5 minutes or so?
5 minutes, ma'am.
Okay, 5 to 7 minutes. Thank you so much. Shall I?
Okay, ma'am.
Okay, I'll just very quickly take you through these slides. So just keep changing. So we have these recent advancements where we primarily have the albumin nanovectors, virosomes. Next one, please. Nanoshells and nanobubbles. Next one. You just enter, I think you can appreciate the movement of the nanoparticles. The nanoparticles, nanoshell, you can see, just press the enter so this video will work. I think you can see the nanoparticles reaching to the target cell. That's how the nanoshells work. Then we have the next slide. Just keep on moving the slides. I'll keep talking about them. So we have some of the diagnosis based on nanoparticles. You can see that we can use the various technologies like fluorescence. In the first section, you can see that gold nanoparticles have reached the tumor and I can appreciate the green color and helping me to diagnose. The next one, please. You can see that there can be selective cytotoxicity based on the types of the cancer cell lines I am taking. I can actually have different types of my nanoparticles, which will give me the various types of this cytotoxicity. Based on this the response of the cell lines, I can select which one should be taken further. Next one, please. This we can skip. With this, I have talked already. Then the we can have customized systems for the breast cancer. Next one. We can have customized systems for the localized delivery of the cervix cancer, because cervix cancer need not. Why do we give it through IV? We can just make a nano drug delivery system which can deliver loco regionally, and we can actually have the cancer treatment right there. The next one. Similarly, the nanomissiles for the ovarian cancer, which needs to be given through the intravaginal route, and finally they reach the ovary and not unnecessarily having the non-site side effects. Next one. The nanoparticles, again for the other systems, can be devised. Next one. The drug delivery for the uterine cancer, each one can be customized based on what the requirement is. So we understand where to deliver, how to deliver. Next one. Yes, you keep on, you can just take through the slides very quickly. These are some of the research papers, just one after the other, which we have produced in our lab. Some of the papers which talked about the various types of. The next one I would like you to just click. This is something, I think I will stop my presentation here. And this, we have many more slides, and that would take a lot of time. It's absolutely fine. Inshallah, next time we can connect again and maybe deliberate further. I would leave the audience here with this particular work which we are doing. You can just finish it off. It's okay. We can just disconnect after this, because there will not be much time to discuss about this. So these are some of the formulation aspects, the way we discover a particular drug, how it is made into a formulation, tested in the animals. We have done this in our laboratory, and we could see the tumor regression. This is my PhD student working on that, and how we reduce the tumor and by making the nanoparticle. The next ones just keep on changing. I'll keep on talking about it. We could go in for the change in tumor volume. We could appreciate that. Next one. The percentage body weight, hemotoxicity studies were done. So this was a research work which I would have loved to discuss. But anyways, then we are able to produce a paper from this. The next thing, primarily, carry on, dear. So this is another papers. So these are some, we can stop here because otherwise it is again another section. So thank you so much for all the patience and bearing with. So these are some of the new works which we are doing in our lab. This is my group, a long, big one, mashallah. Each one of them have contributed. And perhaps you are most welcome to write to us and maybe take any help on any of these things. Next time, if I get a chance, I'll make a presentation specific to Unani formulation, how we have used. This is a slide about Jamia Hamdard. Alhamdulillah, we are having a huge sprawling campus of 99 acres, and which primarily have got schools to the tune of 14. And we are NAC A plus, mashallah, and a school of pharmacies, NIRF rank 1 for the last 6 times. And Alhamdulillah, we are doing a lot of good research on the various aspects of nanotechnology. We are perhaps the pioneers in the field, perhaps the references. And the last slide, you can just take it forward, dear. Thank you so much. My email ID and everything is reflected here. You're most welcome to write back. I might delay the response, but inshallah, my koshish is that I always respond back. I take up calls. So a WhatsApp message is what you need to do if you need to connect me for any of these things. And thank you so much. I would have loved to speak a little more and I apologize for all the hiccups which we had during this beginning of this session. Nonetheless, I trust that it must have given you a smattering and a little bit of knowledge about what nanoparticles and nanosystems are all about. And I'm sure that you can extrapolate this whole thing to your own research, and maybe we can collaborate and do much better work as far as traditional medicines are there. Thank you so much. And I feel bad because I did not do justice to many of the slides, but nonetheless, I hope I have left you with good message about nanomedicines, and we can always take it forward from here. Thank you so much, Dr. Arshia, for all the efforts and being very, very patient.
While still in the experimental stages for many of as a tool for modern therapeutics, potentially revolutionizing how cancer is treated and managed in future. Thank you, ma'am, for your extensive presentation on onco nanomedicine. We are overwhelmed with your in-depth knowledge on nanomedicine and hope to listen more from you in future. Thank you once again.
Thank you so much. Jazakallah khair. If
Now,
there are any questions, if there's time, I can answer.
there's a
I
question,
can,
ma'am,
we can
from
take
one
a
of our
couple of.
participants. Just one question
Hello,
will
ma'am.
take up from one of participant. What is the financial aspect of nanoparticle medicine? How herbal medicine of Unani medicine like decoctions or tablets can be given in the form of nanoparticle?
Wonderful question. Financially, it would be a little taxing because it will require a lot of choice of polymers, which these polymers needs to be sort of like little expensive, and they need to be biodegradable and biocompatible. And as far as your usage of Unani medicines are there, not all the nanosystems would be suitable for it, but I have a lot of hope from this nano emulsions. So we have also prepared a couple of nano emulsions which were based on this Unani systems. So the tablets per se cannot be changed into nano emulsions. Nano emulsions, you will have to find out the active ingredient. For example, if there is an oil which you think is primarily having some effect on alopecia, so you can convert that oil into a nano emulsion, which will maybe improve its penetration and which will be a better option as compared to oil. Similarly, we have done our trials on creation of nano emulsions for treatment of arthritis, where various Unani preparations were converted into nano emulsions, and we found out that primarily this penetration was much better. You may Dr. Arshia, you may share my mobile number if you wish to, so that they can be in touch with me if they wish to. And thank you so much, Jazakallah khair, for all the efforts. Jazakallah
Thank you very
khair.
much, ma'am.
Dr. Steele is going to connect?
Yes, ma'am. I welcome back Dr. Amy Steele, ma'am. Hope you have better connectivity now. And ma'am, you can please share the screen and continue your deliberation.
Absolutely. Can everybody hear me okay?
Yes, ma'am.
How are you, Amy? Amy, this is Zeenat this side.
Yes, I can see you, Zeenat. I've enjoyed your presentation. It's full of rich information as is usual for your talk. So it was wonderful to hear. Thank you.
Okay, thank you so much. I'll be listening
okay.
to you as well. Thanks a lot. That's a pleasure.
That's okay. So I know there were issues with my presentation, and I don't want to take up too much of everybody's time. So I've just taken a few slides back from where it stopped. No technical issues now, is that correct? You can hear me okay and everything? Is that fine?
Yes, ma'am, you can continue.
You're absolutely audible.
Oh,
Amy,
wonderful.
You're absolutely audible.
That's wonderful. Thank you so much. Okay, so I was explaining before my before I took my break with the internet issues, that one of the things that we've generated, and it was so wonderful having my presentation alongside the inspiring work of Dr. Zeenat, because this really talks about how we bring these two things together. The presentation around the things like nanoparticles in the context of something like Unani medicine, and bringing these two ideas together, and not just nanoparticles, but all areas of emerging health technologies. So the site framework was developed as I was explaining earlier, based on a literature review that we did to understand how people were taking traditional knowledge and traditional medicine from in all sorts of areas, and how they were adapting it into modern day, because we use our traditional knowledge and traditional medicine in all sorts of ways. And of course we try and keep the link to the traditional practices, but also there are all these new opportunities and emerging technologies that we can make use of to try and test new ways of expanding and evolving the traditional medicine. Because also, as I mentioned earlier, that these medicines that we use are not they're not static, they're not in time, they're not just something that sits in a book. These are live practices, these are living traditions. They're not historical traditions and historical practices, they're living practices. And so as a result, they evolve and change in our modern day in response to the changes that we have in the society and the technologies that are around us. So the SITE framework is designed to help support our ability to do that in a really thoughtful way, to bring traditional knowledge and traditional practices together with contemporary opportunities and technologies, so that we can integrate those where appropriate and be really conscious of how that impacts and what it might do. So there are 3 sections to the SITE framework. First section is the guiding principles, which are an overarching set of principles to keep in mind when in dealing with any kind of traditional knowledge use in contemporary contexts. And then there's 2 sets of criteria for critically appraising the traditional knowledge before you use it, and then application criteria to guide the implementation of that knowledge. So I'll overview the first 2 more briefly and then spend a bit more time on the 3rd criteria. So within the framework itself, which you can access from that QR code in the bottom corner, we have each section, the guiding principles and the 2 sets of criteria, have a header of their name, and they have a statement or a question that describes and provides a bit of a stimulation for how you might apply or consider the principle or the criteria. And then there's more of an explainer that describes some of these, the information in more detail to help you to understand it a bit more. So within the guiding principles, there's 5 guiding principles. The first is that we have to be accountable in how we translate this traditional knowledge. And we haven't been doing enough of that. We take traditional knowledge, particularly by we, I mean, researchers and policymakers, we're drawing on traditional knowledge to inform decisions and policies and things like that, research studies, treatments that we might be testing. But we're not being as accountable to how we've picked up that knowledge and made use of that knowledge as we justify the background research that we might base our decisions on. We're not justifying the background traditional knowledge in the same level of detail and giving it the same level of respect. And so we need to be more accountable to that. But we also need to be, it's also important that we assess the traditional knowledge and make sure that it is actually traditional knowledge and not, I mean, I looked at some of the studies we found in our systematic review. Some of the researchers came not from traditional medicine background, and they were really just picking up books from local stores that were not traditional medicine texts. They were written by someone who may or may not have had expertise in that traditional medicine, and they were basing their decisions about what they were going to test on things like that, on that sort of resource. And so they didn't make a proper assessment as to how reliable and valid is this as a traditional knowledge source from that traditional medicine system. Also recognizing one of the guiding principles is that point I made before, is that traditional medicine systems are living systems. And so they evolve and change in living practice. And it's a tension that we have between the fact that we're rooted in tradition, we're rooted in these historical practices and this very strong connection to these traditional practices and where the knowledge and practices come from. But we're still living in a modern day. We're living in 2024 with mobile phones and nanotechnology and all of these things, and that as our practices evolve over time into contemporary society, so does the traditions of which we're using in those systems. Then another guiding principle is that that when we translate traditional knowledge into contemporary context, there's a lot of pragmatic decisions that are being made. You know, when we're testing, when we're using a medicine that has been traditionally used in a practice, in a system such as Unani medicine, or using cupping or anything like that might come within Unani medicine, we're making some compromises just by the very nature of the fact that it's being done in contemporary society. You know, we might be buying a set of cupping instruments which were manufactured in mass production, which might be quite different to the ones that were being used a very long time ago when the practice was first evolving. The herbs that we're using are being potentially grown in a slightly different environment and processed in a slightly different way. And that's okay. We're having to translate these things in a pragmatic manner and understand that we're not expecting everything to be the same way it was when these traditional knowledge first arose, that because it's an evolution of practice in a living tradition. But also that there is that clinical experience is an important part of that evolution. And it is a foundational resource in traditional medicine systems. It's not to be belittled, it's to be acknowledged for its strengths and its weaknesses. And it is something that can't be disregarded entirely as being not evidence-based. So they're the guiding principles that came out from our Delphi study as being, anytime people are engaging with traditional knowledge and trying to apply it in modern practice, this is what they need to, or modern settings, this is what they need to take into account. So from that, I went on to, we developed criteria for how to critically appraise the traditional knowledge sources. And there's three main criteria for that. The first is making sure that the traditional knowledge source, and keeping in mind the site framework was actually developed for traditional medicine systems where the knowledge is in written texts. Of course, it is also passed on orally, but there is a very strong nature of written text within the traditional medicine systems. It wasn't developed with indigenous orally focused traditional knowledge in mind. It was based on a lot of these systems of medicine where people don't need to have any contact with an experienced practitioner to help explain or contextualize the work that's being done. They can simply go and pick up a book and take information from it and start testing it or applying it or making decisions or even using treatments based on that without actually being trained in it. And so we really wanted to make the point that in order to identify that you're actually using a traditional knowledge source, first of all, you have to confirm that it's authentic. And so we've got some guiding questions here. You know, can you actually take the knowledge that you have? Is it traceable back to an authentic traditional source in which the people from within that tradition acknowledge that, identify that as being a reliable source? You know, is it, and also is it, does the available source present original knowledge? Is it the actual original source? Or has someone translated it or summarized it or collated a few different sources of knowledge together? And that's not bad or wrong. It's just recognizing that there is some weaknesses or some changes that can happen in that process, and just being aware that to look for how those changes may have occurred. Equally, understanding how the knowledge itself may have been affected during that translation. So the second criteria for critically appraising traditional knowledge sources is how consistent is the evidence across sources. And so one of the recommendations from our experts from our Delphi panel is that if you're looking for some knowledge to understand some traditional knowledge that's in a number of sources, is it actually documented in the same way across those sources? Is there discrepancies or differences? And it's okay, again, if there are, but just recognizing what they are and how that might impact on the accuracy and the usability of that knowledge. And lastly, the safety of the traditional knowledge. And that safety is not just life or death safety, but also cultural and social safety. And other things that are happening now in modern society that may affect the actual safety of that drug, you know, of that treatment, whatever it may be. It could be anything from one of my students looked at naturopathic texts and treatment for women's reproductive conditions. And one of the recommendations that they had for that was that one of the old naturopathic texts was a treatment for that was simply this was to become pregnant. Now, obviously, that may be quite effective in stopping the symptoms of endometriosis, but it's not actually socially acceptable to be a curative response to a condition like that in modern times. Equally, the reasons why people are developing diabetes in a highly processed food environment and a completely different sedentary environment, exposed to a lot of environmental toxins that didn't exist hundreds or thousands of years ago, may mean that some of the medicines that were used to treat those conditions may not be as effective or may have a slightly different outcome as a result of that process. And so these are the things to take into consideration when we're looking at a traditional knowledge source and thinking about its translation into modern contexts. But what I really want to spend a bit more time on, and I am conscious of the fact that time has gone over, is this third section on criteria around the application of traditional knowledge, because this is particularly where the concept of emerging health technologies and its integration with traditional knowledge really comes into play. So there's 8 criteria to think about in the application of traditional knowledge in contemporary settings, contexts, and practices. And I really want to emphasize that not a single one of these is meant to diminish or devalue traditional medicine and traditional knowledge. In fact, it's supposed to do the opposite. It's supposed to give us a framework to have a mature conversation and engagement with traditional medicine and traditional knowledge, so that we can recognize when we're adapting it into contemporary settings, because by and large, we all have to for a number of different reasons. What does that mean? And how does it have an impact? And giving us a list of things to think through so that we can be really conscious about the potential difference, that when that documentation was first put together, when the expectations that were documented around that treatment or practice was first written down by whomever it was of our traditional medicine forebears, that the outcome we see today may be different for all sorts of reasons. And the way that we apply it today may be different for all sorts of reasons. And just for us to give an opportunity to think through some of that. So the first thing that we think about when talking about finding a traditional medicine piece of knowledge and wanting to apply it today in research, education, policy, or practice is, to what degree is the way I'm trying to apply it align with the core characteristics of the tradition? So if we're trying to adapt traditional medicine knowledge into an emerging health technology, and I will, just because you've just had this wonderful presentation from Dr. Zeenat, I will draw a little bit on nanotechnology as an example, because it's so perfect, is being able to identify to what degree is the traditional knowledge being applied in a manner that aligns with the core philosophical and practical characteristics of the tradition. And so there's one thing is to take a herbal medicine and to apply it to use nanotechnology to improve the delivery. If you're it's one thing if you're using it to improve the delivery in a manner and for a condition for which it would already be used. You're just trying to improve the delivery, and you get a great example of, using some nanotechnology to improve an oil application of a herbal medicine to cover across the cutaneous barrier better. So this is a really great example of how that is just improving and optimizing the application. Very different to someone taking it really out of context and trying to deliver it in a completely different way through oral administration for perhaps a herb that's always been used topically, for a condition that it's perhaps never been used for. And keeping all of that in mind is really just recognizing that, again, it's not wrong to try a new delivery method or to try it in a new condition, but how does it differ? And how much can we then rely on that traditional knowledge if we've moved it so far away from where it was? Or how much do we have to do additional research to check its safety and effectiveness and other such things? So really keeping in mind that for it to be truly a measure of traditional knowledge, then we need to think about how much is it actually being applied in a manner in which that traditional knowledge first arose. And another criteria for thinking about the application of traditional knowledge is person-centered translation. And again, this is most traditional medicine systems, and Unani medicine is definitely within this group. They have a very core focus around person-centered care, user-centered applications, holistic approaches, and individualized clinical application. And again, if we're moving away from that approach and be developing more algorithmic and generalized treatment that's not individualized, then we have to recognize that this is not the way that traditional medicine system was designed. They weren't just using the same drug for the same person or for the same condition all the time. There was lots of different mixes of treatments, people, lifestyle and diet and herbal medicines and other, body therapies were all being combined into the same treatment. It was complex and it was individualized. So those sort of combinations are really things for us to think about when we're taking something out of that person-centered, complex and holistic application. And just being aware that that may change the outcome as a result. The ethical applications or ethical approaches or considerations is another part. Is being aware of how the intellectual property part plays a role in how we're applying it, but also that potential social and cultural impacts of implementation, and that kind of speaks back to the sort of things I was saying before about some of the treatments and recommendations that may have been acceptable hundreds or thousands of years ago may now longer considered to be unacceptable today for social and cultural reasons, not because they're physically unhealthy, but because they have other social and cultural impacts that are considered unacceptable. A fourth criteria is the tradition-informed communication and framing. And this is a really interesting one that the experts brought forward. It's really important that we find a way that doesn't completely strip away the traditional philosophy and principles when we're talking about the traditional medicines themselves. And that's a really difficult thing to do in the context of emerging health technologies, when we've got such a clash of worlds almost of this, digital and, technologically advanced practices and tools and instruments being delivered and employed alongside this ancient traditional medicines, but making sure that in the process of trying to communicate the traditional medicines to all of the for all of this, to whoever it is the audience may be who's not from the traditional medicine, that we find a way that doesn't completely strip away the all of that context and important language that goes with it. Another criteria for applying traditional knowledge in contemporary settings and emerging health technologies is really being clear on the accuracy of the interpretation of that traditional knowledge. And certainly when we're picking up traditional knowledge, there was a lot of terms that they use, as I'm sure many of you would know, for health conditions and such things that have changed over time. I mean, in anglicized traditional medicine systems, they talked about dropsy instead of congestive heart failure, or things like that. Sweet urine disease or sugar disease for diabetes. They're just small things. But there's lots of different changes to the way that languages, that conditions are being discussed, but also, and so therefore for us to make sure that we're actually applying them in the same condition, in the same way, we're really aware of how that translation of terminology has occurred. And equally that any of that translation across language and also across culture. And certainly for Unani medicine, which has evolved from Greco-Arabic in Indian and into India, it's got a very strong role in Indian medical landscape nowadays, being aware of how that cultural adaptation may have occurred over that time, and just how the potential areas where errors in translation may take place. And particularly if people are coming in who haven't been steeped in the traditional medicine system and who are drawing on that knowledge, making sure that that's most at risk, because they wouldn't have any of the nuances that would come with the training of being actually trained as a practitioner of that system of medicine. They might just read a piece of text or read a document somewhere and misinterpret information because they don't have all of that nuanced cultural context that you get trained in when you're trained as a clinician. So, and the next application criteria is about transferability to the contemporary context. Is it appropriate? To the contemporary setting? Is it appropriate to adapt that particular medicine using nanoparticles? Is it appropriate for us to develop this particular diagnostic screening method that uses a combination of the Korean Oriental medicine groups have done amazing jobs with doing some of their constitutional diagnostic skills using technology, facial recognition technology and machine learning and AI. And they've done some really amazing things with that. And it's just having that moment to stop and think, is it appropriate? Is it make sense practically and culturally for that to work? And in many situations, the answer will be yes, but it's important that we stop and ask that question. Can the traditional knowledge be adequately adapted while still being safe, effective, and having integrity to the philosophy? So I think having that question is really, that comes back to a lot of the points that I've been raising. So apologies, my speaker's just changing over. Is what will the what do we lose or what do we gain, and what impact does that adaptation have as we take something that was developed in very different circumstances with very different technologies at the time available and is being used in incredibly different ways today? And how much does that impact on the potential safety, effectiveness, and the alignment with the philosophical integrity of the traditional medicine system from which the knowledge has arisen? There's also the point about the accessibility and integrity of traditional resources. And this is particularly, oh, did I lose my presentation? No, I didn't. Sorry, I've just lost looking at my presentation. I don't know why that happened. Oh, there we go. Okay. So the accessibility and integrity of traditional resources is another part. And that's really just asking the question, do we still actually have access to the right materials as they would have used back then? How important was it when they produced and manufactured a particular therapeutic treatment? They made a herbal remedy up and they may have maybe prepared it in a copper pot and stirred it for 2 or 3 days and done certain practices in the preparation that ethnopharmacology is doing a really great job of exploring some of that. But, how important were those materials, equipment, and facilities? Do we still have access to them in a meaningful manner? And are the preparations that are available today, if they're that very different from the way they would have been prepared back then, how much can we expect, based on all the things I've considered so far, that might impact on the outcome from us using a similar but not exactly the same preparation as what it used to be? Can we maintain the quality and efficacy of traditional knowledge and those resources as we adapt and change and implement and adopt these amazing new opportunities that the emerging health technologies are giving us? And where is the balance with all of that? And I guess the last thing, the last criteria around the application of traditional knowledge is what is a genuine need that may be addressed by the traditional knowledge? And, can we, you have to really think about making sure that there is that comparative benefit between traditional and non-traditional approaches can be put into place. So, all right. Apologies, my slides are just stuck for some reason. There we go. So that's the overview of the EPIS framework. And one of the things I wanted to point out, which I mentioned much earlier, but I'm not sure if my audio was causing problems, is when we look at it across the EPIS framework, when we did the literature review of what had been reported so far and how traditional knowledge was being used to inform contemporary research, education, policy, and practice, is so much of the description of people using traditional knowledge centered around the exploration and preparation stages of how did they identify the traditional medicine knowledge that they were going to use, and how did they make some choices about how they were going to turn that into a contemporary treatment or practice. What the EPIS framework does is not only does it address those first 2 stages, but it really provides some strong guidance for the implementation part, the sustain and how do we sustain that treatment in practice, and thinking through all of these nuances and these complex issues that we need to consider if we're going to be bringing traditional medicine knowledge from hundreds and thousands of years ago and applying cutting edge technologies in our implementation of it. So I'm going to provide just a couple of really short examples because I'm very conscious that this time, the webinar has probably gone for longer than you've put the time aside for today, and it's probably heading into your evening dinner time in India. But just a couple of examples. So this is just a couple of studies. One of them, this is what the picture on the left is just some images that came from a paper that's been written about using Chinese herbal medicine for chronic obstetric pulmonary disease. And as you can see, there's photos of the specific plant. There's the plant part as it is used in the herbal medicine. You know, they're really dealing with the whole plant extract and extracting it in a certain way in accordance with traditional Chinese medicine. And we compare that to what's going on the right here side, which is taking a lot of active herbal medicines, and some of these are actually used in Ayurvedic medicine. I'm not sure if they're used in Unani medicine. I do not profess to be an expert in Unani medicine. But you can see this is much more focused on, it's acknowledging the plant that they've come from. They're focused on the active ingredient or one of the active ingredients and really getting into the details, the minutiae of how that might be working in the body, looking specifically at nano-delivered technology, in fact, in this instance. And I guess the question that we ask from that is just going back to those examples that I gave before, you know, the question of accessibility and integrity of traditional resources. Are the necessary materials and equipment and facilities available in order for these plants on the left to still be used today? Are there issues around the sustainability of those plants? Are we able to reliably access those plants in a way that we need to? But equally, there's questions around that person-centered translation. And as we get further and further into the smaller and smaller details of the specific plants, which is such valuable knowledge and so powerful for us to be able to wield that as a tool. Also keeping in mind that parallel question of, is the traditional knowledge being applied in a manner that's consistent with person-centered care and user-centered applications? And somehow, as people who have a foot in the world of traditional medicine and in this case Unani medicine, and people who also have a foot in science and in research, realizing that somehow we have to be the leaders in bringing those two thoughts together. The idea of exploring the specifics of how things work and how we can make them work more powerfully, more effectively, and in a more targeted manner, whilst not losing sight of the human in which that's happening, and the humanity and the holistic requirements of a person who is experiencing the health condition that very targeted treatment is focused on. Just as another example, and it was beautiful to the example around the oil that was given earlier, because it really related to this. Again, we're looking at delivery methods, and I honestly didn't realize that there was going to be that talk on nanotechnologies when I put this together. But it's just these studies are based on one of the researchers in our group, Kamal Dua, who does a lot of pharmaceutical delivery method research for herbal medicines. And, you know, when you look at this process of taking a herbal medicine and converting it into copper nanoparticles, and then, you know, identifying its antibacterial and antioxidant activity, we think about that translation of this raw herb that has been used for hundreds or thousands of years for a particular purpose, going through this very modern technological adaptation to improve its delivery and check on its activity. And I think just making sure that work is coupled with the question of, can we do this while retaining the safety, efficacy, and the philosophical integrity upon which this plant was originally designed to be used, and which we're building on all of that knowledge. We shouldn't throw it away in the process. We need to find a way to wrap up all of that traditional knowledge and expertise in the rich history of that plant's use when we're developing new technologies and new applications. Equally, when we're looking at different delivery methods, and this is just an example again from some of Cam's team that's looked at topical creams and hypodermic needles and microneedle patches and transdermal patches, all these different ways of delivering giving topical applications and how they can reach different areas of the dermal layers. And just coming back to that question of, is the traditional knowledge being applied in a manner that aligns with the core philosophical and practical characteristics of the tradition? As we're thinking about, the different ways we want to apply a herbal medicine, a topical herbal medicine or a tropical treatment of some kind, and we're considering these different wonderful technologies that have been developed that help the application, really just keeping in mind that we need to ask and answer this question. And I just want to reiterate a point that I made earlier, that none of this is intended to either make traditional knowledge wrong and bad or emerging technologies wrong and bad. It really is just designed to give us a framework to have a really thoughtful engagement with these next steps forward as we find a way to meaningfully and effectively, powerfully embrace these new technologies whilst respecting and honouring and working with building on the traditional medicine knowledge systems from which we have got such a wealth of opportunity in those areas, and making sure that they are working together as harmoniously as possible, and that every decision that we make as we integrate them together is done in a way that is thoughtful and respectful of the opportunities of both. So this site framework has, a number of areas of significance. It was developed through collaborative participatory research methods. It was through an in-person stakeholder forum, a global Delphi study, and it was really developed with the traditional medicine research, policy, practice, and education community. It's really designed to, in the face of evidence-based practice and emerging health technologies and evolution, maintain the integrity of the traditions from which traditional knowledge and traditional medicine is based and arises and still exists, as well as recognizing the intellectual property of that work. It's designed to support the use of approaches that are tailored to traditional knowledge, and allows us to also look at ways that we might adapt these emerging health technologies and to make them more suitable for traditional knowledge, rather than always just thinking about how we can take traditional knowledge and apply emerging technologies to them. There's a lot that I think that we can benefit, that emerging technologies can benefit from when we start to think about these traditional knowledge challenges, because they start to adapt through that process, and then we end up with new opportunities that arise. And the other thing about this is it's really designed to support appropriate and ethical evolution of practice. And that practice is in all of these contexts and settings. And as I said, the idea of it being conscious and thoughtful, and for us to treat traditional knowledge with the same maturity and respect with which we treat research-based knowledge is really what our team want to see come out of the site framework. So the site framework is already being included in curriculum of traditional complementary medicine educational institutions in different countries. There's uptake from leading TCIM journals for them to include, for people to include more detail around the traditional knowledge and how they've justified and employed the traditional knowledge in their decisions when they're testing new treatments and practices. And it's also being integrated into, and considered in evidence-informed policy-making process, opportunities and activities that are linked to the World Health Organization and others as well. So, and also in the Food and Agricultural Organization have recognized the potential opportunity of the site framework. So, thank you very much. I'm appreciative of the time. I'm very conscious that this is much later than you probably had originally intended, and I'm very grateful for being invited to share this, the framework, and particularly to be a part of this conversation about such an important area for our field, because you know, we have much to learn, much to offer, and much to benefit from emerging health technologies and traditional medicine systems. So thank you very much.
Thank you, ma'am, for a comprehensive presentation on emerging technologies for traditional medicine. You covered from the review of literature to taking us through the steps of EPIS framework, the SIDE framework with their significance and impact. In the world increasingly driven by the digital innovation, this framework emphasizes the need to respect and incorporate various systems of knowledge, including indigenous and holistic approaches into contemporary healthcare systems. Thank you once again, ma'am.
Thank you.
Ma'am, there's a question from one of our professors. How far we compromise holism for explaining our concepts by reductionist model of knowledge?
Well, this is, I guess, part of the point of the SITE framework isn't to answer all the questions, but to identify what questions need to be answered by everybody. And I think that that's an interesting conundrum that we face in terms of how do we describe holism to people who think in a reductionist manner. One of the things our research team has done is we've started to use complexity science modeling, and we've captured, and I'm happy to share some of the papers that have been done around this. We've used naturopathy as a case study, but we've captured clinicians' clinical reasoning in how they make sense of a case, and we've actually mapped that in accordance to complexity science to show this holism. So not just about how complex the treatments are, but how complex the thinking of a clinician who applies traditional medicine practices and who's embedded and immersed in traditional medicine philosophies will actually apply this to their way of interpreting the case. Because we don't even decide on the treatment until we've actually made sense of the case itself. And once we've made sense of the case, then you end up with a treatment. And it's that's a big part of what drives traditional medicine philosophy. And this is one of the reasons we're saying we can't just start thinking in a reductionist way where we start thinking, oh, there's a treatment for that condition, because that is not how traditional medicine practice was ever done. It was never done in that way. It was always very complex in the thinking of the clinician itself and their clinical reasoning. So I think trying to find some of those methods, some of those more recent scientific methods that recognize complexity, and using those to somehow capture and describe what it is that we do, that doesn't diminish the other work as well. And I'd be happy to share some of that. And I'm sure that's not the only method with which we can do it, but I think trying to use the benefit that we have today that we didn't have 20 years ago is that science itself is recognizing, particularly medical science, is recognizing complexity and the and the role of complexity in all of the treatments that are being done. And so we can use some of those developments that they've got to help decode for them the language that we've developed over time and the thinking and the approach that we've developed over time, so that we can create a bit of a language bridge through using those methods.
Any more questions? Can we take few more questions, ma'am?
I'm fine with that. I'm here, so that's fine.
Any more questions?
I can see there's a couple of previous questions as well. There was something about
Ma'am, sir is asking about how do mixed methods of research help traditional medicine?
Mixed methods that is very, very important, and they're in both helping to understand the outcomes of the treatment, but also helping to understand how we're going to go about measuring the effect of our treatments in the first place. I think that the mixed methods is not just about qualitative focus groups and interviews and those kinds of things as well. We've got some of our researchers have done mixed methods analysis of traditional texts as part of analysing the traditional knowledge and helping to make sense of that traditional knowledge. So they've done, so there's lots of different ways. I genuinely believe, and our team does a lot of mixed methods research and not always publishing it as mixed methods. Right now, I'm, writing up a survey paper on some of the work that we've done, and we've also done focus groups that have led on from that, and so the project has been conducted in mixed methods way. So mixed methods is very, very important, particularly because in traditional medicine, or in all healthcare, to be honest, patients aren't always motivated by what motivates doctors. And so for us to capture some of those other nuanced changes that happen that we weren't even expecting to happen when we set up the study in the first place, to capture those experiences that patients get from the treatment that weren't the things that we were expecting for them to come out. We need the qualitative work to be able to follow up. What did they experience? What did they like about it? What didn't they like about it? What would they like to see different? That work is really best done through qualitative research, and it complements the quantitative hard data really beautifully.
How far content analysis will help in such scenarios?
Well, the way that the content analysis helped in this situation, the student did a phased project where she engaged with traditional texts, and she was able to actually map the evolution of traditional medicine practice and knowledge. So this is actually, in the one that I'm thinking of, is the naturopathic study I was talking about in terms of women's pelvic pain and reproductive health problems. And she evaluated the traditional texts of naturopathic related to women's health for naturopathy, and she pulled out all of the knowledge that she could find in this set library of traditional naturopathic texts, and did a content analysis of what was covered, what treatments they were recommending for which conditions, how frequently they were occurring, so we could get a sense of consistency, and those kinds of things. She then did a similar content analysis from texts that were included in contemporary naturopathic curricula. So some of those would have been referencing the old texts. A lot of them were based on, a mix of old and science and clinician experience and other such things. And so she then compared how some of these practices had stopped, because certainly some of the things, and I'm sure Unani has similar as well, some of the treatments from the old naturopathic texts we know are no longer any good. We no longer prescribe mercury to people. It's not something that's done, where it may have been done a number of hundreds of years ago. It's certainly not done today. And so there are certain things we're able to show are no longer in practice, other things that had evolved into naturopathic practice that weren't there before. Certainly, as some of the amazing research that's done on some of the medicines that have come from other continents and traditional medicine systems, naturopaths have started to pick some of those up, and so that was showing up. And then as a third part of that study, she then surveyed contemporary naturopaths who specialize in treating these conditions and asked them about what they think is the best way of treating these conditions. And so she was able to map the changes that were happening from the content analysis and the survey research across time. So I'm not sure if that genuinely answers the question, but I think that's an example of how content analysis can be used in this work.
Can we take a few more questions, ma'am?
If you have any more questions that you'd like, sure.
Sir, different philosophies emphasizes specific dimensions of the problem. So how to systematize such things, ma'am?
That's an interesting one, and I think that's a question. Do we need to systematize that? I guess, I think that there's a philosophical question in that do we, the different dimensions that are associated with the problem need to be recognized. Certainly what we need to do is start documenting that. And as I said, one of the biggest issues I think we have with the way traditional knowledge has been drawn upon for research up until this point is it's usually a footnote. And what I mean by that is if someone does a study, it's maybe a clinical trial using a herbal medicine, or it could be a drug discovery study or whatever it may be. Any reference to the traditional medicine roots of that treatment is usually found in the introduction of the research paper. It's usually flagged in that as this has come from. It's commonly used in traditional Chinese medicine or Unani medicine, and it might even at a stretch mention the traditional medicine text that it is mentioned in, but it's never mentioned in the methods section. They never systematically go through and say, we looked in these texts and we found this information, and of that information, we made a decision based on this inclusion criteria that this is a traditional medicine that we are going to pursue and study for this reason. That's the systematization that I think is desperately needed for traditional knowledge. The process, we would go through, do exactly the same thing if we were doing a drug discovery process involving existing research. If we wanted to do a clinical trial, we would have to show that there was animal study research, that there was safety research had been done, that there was in vitro, in vivo justification. And it would sit in the introduction, but at the same time, there would be elements within the methods that showed that that the justification for how this choice was made of which was what was going to be studied. And this is the systematization that's needed more than anything else, I think.
Hello, ma'am.
Yes, hello. Someone's got their hand up. Is that you?
Ma'am, how can artificial intelligence can be used in traditional medicine?
That's a really great question as well. And I'm not an expert in artificial intelligence. But what I will say is that where I see lots of opportunity with artificial intelligence is potentially in trying to engage with some of these, as I said, Korean Oriental medicine. There's some really great work that they're using with facial recognition technology as part of their constitutional diagnosis methods. And there's actually apps that are now being developed from Korean medicine because they use the facial recognition, the facial morphological features as part of their diagnosis of constitutions. And so AI can definitely be used for those kinds of things, and probably to good effect as well. And certainly people are also using AI to look at some of the network pharmacology of some of the traditional complexes, and certainly a lot of traditional medicine systems and have established complexes where the same herbs are combined to achieve for certain reasons, and those combinations have got traditional names that are used. And so understanding the interaction between these different processes, it's much easier. Well, the AI has the capacity to distill down some of that complexity in a way that people who are trained in a reductionist manner can start to understand it. It can bring it down to that point. And certainly we can use it to help to understand. I've got a PhD student at the moment who's done a clinical trial using individualized herbal medicine, which allowed the clinicians to prescribe the herbal medicines that they would usually prescribe for that individual, but they were all treating depression or anxiety. But they were given free rein to prescribe any herbs that they would wanted to for that individual based on the clinical case. And so we'll be able to use AI technology in our analysis to look at the relationship between the different herbs that were prescribed across these 250 odd patients to see if there's particular herbs that had particular benefits when used in combination with one another. And so we can use it to evaluate some of that complexity in a way that would just take too long for the human statistical models to potentially do, or will take longer anyway, not take too long, but take longer. But as I said, I'm not an expert in AI, and what I do know about AI is that it is definitely as good as the people who program it. And I'm sure there'll be more and more opportunities that arise over time.
Thank you so much.
That's okay.
And how can we address positive indicators of health in traditional medicine?
I'm not sure. Can you ask that question again? I'm not sure what that question is referring to.
Oh, yes, please.
Sir, kindly address it.
Ma'am, I had two questions. One was primarily, are we able to address supramaterial concepts in science? Usually traditional medicines, they deal with certain concepts which have basis in supramaterial concepts, like we say the soul or pneuma and other things. which we are very much, it becomes very much difficult to explore it in terms of scientific parameters. Secondly, there are certain positive indicators helped in traditional medicine. But usually when we see it in the present conventional medicine, all the indicators are absence of disease or negative indicators. So how can we try to bring it in the mainstream medicine that there are positive indicators also?
Right. Thank you very much for clarifying that. That's really helpful. Look, to be honest, on the supramaterial concepts and science, I don't have an answer for that. I don't. And what I do think, and I guess part of what the site framework is proposing is that at the very least we need to recognize that we're not measuring it, and that it is part of what's considered important in traditional medicine, and it's going unmeasured. And that just at least recognizing that rather than skipping over it and pretending like it doesn't exist, and that at this stage maybe we don't have the methods. We may well have. It may be in quantum physics, and it's just not my area. I'm a public health researcher, but I think that is an important question, and it's just acknowledging that there are elements that are recognized as being really important to health within traditional medicine that we cannot currently measure, and that may mean that there's an element that is unevaluated, or that we're not properly describing the true benefits that as seen and understood by traditional medicine. And then that's okay. We just need to recognize that and take it into account when we're measuring the outcomes of the treatments or whatever it may be. So, and in terms of addressing the positive indicators of health, so the wellness orientation, salutogenic orientation of health in traditional medicine, I think that there is actually more and more opportunities. And look, they're still limited. But I think one of the things my first thought is there are some instruments now. There's a personal well-being index, for example. It's a validated instrument, and it measures somewhat more holistically. It includes the sense of spirituality and things like that. And it's somewhat more salutogenic or wellness-oriented rather than illness-oriented. It's not, it's not like quality of life. It's more about, do you feel connected socially? Do you feel like you have a really full and happy life, and those kinds of things? And so it's a personal well-being index. It's a validated instrument. We use it, and it's not been developed by traditional medicine either. It's actually been developed within health promotion and public health, and it's recognizing that there is there's a sector of the health research and health practitioner practice and health promotion community that are trying to achieve wellness, not just absence of illness, and that we need to be able to measure that. So the good thing is that some of that work is being done, and we can draw on it. But the other part is, if there are things that we feel aren't being measured properly, such as wellness orientation, that's where people like our group and others who are working more on that public health side of things, and even psychology research, but that we have a responsibility from within traditional medicine research to develop instruments that do measure those things. We can't sit and wait and complain that those things don't exist. We have a responsibility. If we feel that the instruments that are available beyond the supramaterial concepts, but some of those very fundamental wellness-oriented positive indicators of health, we need to be able to develop instruments that capture and define those things ourselves. And one of the things I always encourage from I've actually got a PhD student at the moment it's astounding when you start looking at it. His project is going to look at how practitioners, health professionals generally, but focusing on traditional medicine practitioners, determine if their patient's getting better. How do they just define clinical success? Now, we thought we would build off how biomedical practitioners determine clinical success and use a bit of a framework and then adapt it to the traditional medicine context. Turns out there is no framework for how biomedical practitioners determine if clinical success. They don't have one. So it's astounding to me that after all of the money that's spent on biomedical research, we still don't know how doctors decide that their treatments are working. And so what we start doing is we develop those instruments ourselves from within our team. We use the case study of traditional medicine, but we write them for health. We make it available to the wider health practitioner community. We don't make it about traditional medicine. We just use traditional medicine as the case study in it because we believe all of health can benefit from that work. And it is really important work. And it's because we're coming at it from the wonderful question that you've asked, that question of how can we address positive indicators of health? This is coming from that traditional medicine, holistic, wellness-oriented thinking that we are immersed in, day out in the work that we do. And I think when we bring that to our research questions and our research activities and our clinical care, we start looking for things that biomedicine hasn't gotten around to looking for yet. And so we probably have to start creating these things for ourselves.
Thank you, ma'am. Thank you.
Any more questions?
Thank you. Thank you.
Thank you very much, ma'am.
Hello, Amy.
Hello, Dr. Zeenat.
I was sitting through your presentation and wonderfully done. I must congratulate you for that. And of course, I think the last point has to be the one which you just mentioned, that we cannot keep the traditional medicine in isolation. We have to use the modern standards to prove how efficacious it has been, and it is for sure. So, and I was it was very heartening to see the nano parts as well in your presentation. So that's something. The message is very clear that there is no system which is not amenable to Unani system for that matter. Any system can be amalgamated with the traditional medicine as well. And when the need of the time is that we have to give them the data as per the modern protocol, I think we should not hesitate because we have got enough strength in our systems. So that's one thing which is a take-home message from your beautiful presentation. And it's very nice to connect with you. Thanks a lot. I think we must thank Arshia collectively for bringing us together and bringing up this beautiful seminar. And I'm sure many would benefit.
Absolutely, and thank you to Ashia and the team for your professionalism during both Zeenat and my technical glitches as well. You did a wonderful job.
I don't know. It never happens. You are still like so many kilometers away. I was not that far. But it had to happen. But Allah has been kind. We were able to deliberate despite those hurdles. And I think it's the patience of the audience which needs to be appreciated. That they were like stuck in good numbers. So that's something, I think, collective apology from both of us to the organizers for the glitches which we faced. Lovely to see you, Amy. And thanks, Arshia. And thanks, everyone.
Thank you.
Thank you, ma'am. It was a privilege having you with us. Before we conclude, I invite Dr. Sharif Shamsi, sir, Associate Professor, Department of Ilmul Saidala, National Institute of Unani Medicine, Bangalore, to propose vote of thanks.
Thank you,
Over
Dr.
to you, sir.
Thank you, Dr. Kausar. Good evening, one and all join here in the webinar. On a lighter note, I must say here that I hope there will be no technical glitch during the vote of thanks. I want to extend my sincere thanks.
There will not be.
I want to extend my sincere thanks, first of all, to the Director, National Institute of Unani Medicine, Professor Sayyad Shah Alam, sir, for permitting us and providing the best facilities for this webinar. I am very much thankful to our speaker, Dr. Amy Steele, Associate Professor, School of Public Health, Australian Research Consortium in Complementary and Integrative Medicine, Sydney, Australia, who joined us but could not continue her talk in the earlier period due to technical glitch. Her topic was Navigation, Emerging Technologies for Traditional Medicine, a framework to bring together different ways of knowing in a modern world. I apologize that there is some technical issue and voice was not clear at that time. That's why we moved on to second lecture before time. But again, we connected very well after Professor Zeenat Iqbal's lecture. The lecture of Dr. Amy Steele was very insightful, very deliberative lecture, very understandable. And I'm also thankful to our second speaker, Professor Dr. Zeenat Iqbal, ma'am, Professor, Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. She speaks in a very elaborative way on nanomedicine, a putative tool for modern research. Her lecture was very clear and properly understandable. She elaborately speaks on various important subtopics like challenges, barriers faced by nanoformulations, etc. I would also like to thank the organizing chairperson, Professor Arshia Sultana ma'am, and the organizing secretary, Dr. Kausar Fatima Firdaus madam. Thanksgiving lecture is not complete without the name of our IT cell man, Mr. Rafi, who has been regularly involved for the last few days to arrange all the related things needed for the webinar, especially the Zoom. In the last, I am extending my thanks to all the participants of this webinar. Thank you very much all. Thank you.
Thank you so much.
Ami ma'am, I'll take a moment. Participants are asking for your email ID if at all they have a query. If
Yes,
you
I'm
feel
happy
it
for
is-
that to be shared. I'm happy
Can
for
you
that
please
to be
share it in
shared.
the chat box?
Okay, hold on.
Good evening. I'm very thankful to Madam. It's night in Australia, but she's connected with us. And I'm also thankful to Dr. Zeenat giving us so good insight about nanomedicine. We are really enriched with your both knowledges. Especially Ami Madam, she is sitting in night also. I think so it's 9 or 9:30.
It's 10:45 here.
Thanks a lot.
Thank you so much, ma'am, for your time. Hope see you both madams in near future. Listen more from you. I thank all the participants and the organizing team once again. Thank you one and all. With this, we conclude today's webinar on emerging technologies in healthcare. Thank you once again.
Okay, thanks, dear.
Thank
Thanks a
you.
lot. God bless you.
Goodbye,
Allah bless you.
everybody.
Take care.
Take care. Bye-bye.
Delegates can leave.